Buongiorno a tutti. Good morning, everyone. Nice to see you here. I know I'm taking you away from the beautiful outside weather, and it's a great pleasure to be with you here today in Locorotondo. I'm John Paul Judson. I will be your host and moderator for the day. Now, you know, when we talk about health, you know, One Health, human health, animal health, plant health. Usually plant health comes a little bit low in terms of, you know, interest for people and media and public attention. You know, it's something that can happen, problems that can happen in a field somewhere that you don't really see. And it's an economic problem for maybe farmers. Oh, okay, it's their problem, you know. But when you travel around Puglia and you see some of the devastation of the olive trees, I think plant health suddenly becomes a little bit of a different category of issue. And obviously there's the economic issue, but I think what's even more striking is the heritage issue. Because it's like a mark, right, on a region that has been so represented by olive trees, and, you know, we're talking millennial trees in certain instances. So I think if there's anywhere in Europe that has a strong understanding and knowledge of the importance of plant health and how to try and maintain plant health, it's probably Puglia. So it's really a great honor to be able to discuss the issue of Xylella fastidiosa here. I think it is the best place to do this. And, you know, obviously we'll be hearing from a research project, BIOVEXO, which is holding, therefore, its final conference here. And it's good that there is attention on this issue as to how one can try and solve the problem of Xylella fastidiosa. And we will be discussing some of the solutions that have come out of this project, but also throughout the day we'll be discussing the broader challenge of dealing with the disease. So I've also been asked, and I'll say it now, I'll say it also at the end of the event, to please remember when you leave to don't leave with your badge, okay? Please make sure that you don't leave with your badge. I'll say it again at the end of the event, don't worry, but it's subliminal to say it a couple of times, right? So we'll be together for the whole day. I will do my best also to get your views on board, and so I hope that you'll be able to feel free to ask some questions to our speakers. And in order to get us going, I would like then to hand over to some colleagues who will make some introductory remarks, representatives from the BIOVEXO project, but then also from our host institution today. So we have Stephen Webb, who's chairman of the RTDS group. We have Stéphane Compin, who is senior scientist at the Austrian Institute of Technology. I would also like to welcome Dr. Francesco Di Serio, who's director at the CNR Institute for Sustainable Plant Protection. And I believe there is someone called Enza, who will be representing the host institution today. Enza, where are you? Anyway, she will be coming up soon, I am sure. Okay, so I hand over to maybe Stephen first.
Good morning, everybody. So, welcome, and thank you, Jean-Paul. We have a few people still arriving. Of course, everything is still kind of warming up here. So my name is Stephen Webb. I'm the leader, chairman of a small company in Vienna. We are specialized in setting up European research and innovation projects, especially in the field of sustainability, and we're very proud to be coordinator of the BIOVEXO project. So the BIOVEXO project is, for those who may be hearing about it the first time, it's actually been running over 5 years now. We began in May 2020 with the challenge of trying to address this problem, obviously, of the Xylella fastidiosa, and trying to find an environmentally sound way to eradicate the disease and to without destroying the soil or any other parts of the ecosystem. And so this project began in May 2020. Actually, we were here, and I'm sure some of you, I see they're still coming in, which is great. In October 2021, COVID had just finished, and I remember we were sitting here. We had, I think it was one of the first events after the COVID pandemic, and it was really well visited. I think we had about 80 people here, and every second seat had tape on it. It was like just a jolt your memory banks back to those times, every second seat was like blocked off, and masks and all the full drama. But it's so great to be here again in Apulia, that everything is kind of back to normal. Obviously, we're still dealing with our problems here with Sayala. So the project now is just coming to a close. In December 2025, we'll complete the project. It was quite a large project, and at this moment, I just want to also say that we're using taxpayers' money here and trying to deliver results which are important for Europe and for Apulia, but also for other parts of Europe. So we spent. Well, the whole project had costs of about 8 million euros, and we got granted 6.6 million euros. And this is because the companies involved are also investing in the project. It's not, so the companies here, and you will meet them today, have invested in this project because they also want to bring product to the market to solve the problem. So it's an innovation project, which means we're not just doing research, we're trying to develop products and services to actually solve problems. And at this, also, I'd like to thank our funding authority, and that's the CBE, the Circular Bio-Based Europe Joint Undertaking, CBEJU, which is, you might see sometimes it's called BBI as well, because it was before when we started the project, it was called Bio-Based Industries Joint Undertaking. And the joint undertaking means it's an industry-driven program. So it's not purely taxpayers' money. The industry is putting, I think it's about 2 billion euros into this fund as well. So that's driven by the industry and supported by the European Commission. So it's a so-called public-private partnership. And the Bio-Based Consortium is the private part of that, and that's what basically let's just say it's a combination of private sector investment and the drive to find bio-based solutions to improve Europe and in all aspects basically. So yeah, just thanks to our project officer Oliver Sobel. He's not here today. He's in Brussels. You might be following us online. We are having this running online on YouTube, so there will be it'll be able to be accessed also later. So Oliver's not here, but we are very happy to have Karen Niedstadt here from the European Commission, and she's a policy officer, which is probably also much more relevant for you and for us, because we're going to talk about how we're going to actually take the results out of BIOVEXO and put through the regulatory authorization process to actually bring product to market. So Karen will be giving us a talk today on that. So our job as RTDS, we are the administrative coordinator of this project. We lead dissemination, exploitation, and communication, which means about managing results and helping understanding the processes of engaging with stakeholders and making sure that our project results are relevant and addressing the needs of the stakeholders and organizing this event. And part of that job is, of course, just keeping it all together, but also keeping the whole thing running. It's quite a big job to run such endeavors with, it's a lot of people involved. I think we had 11 partners from 5-6 countries. But we've had a good run. The project's coming to an end. We've had, all the partners are here. So do feel free to, of course, approach speakers. We're going to try to make it today a little bit as interactive as we can. We'll have microphones to move around and answer questions, and we'll talk about what we've achieved and who else is involved with this and way forward. So on that point, I would like to hand over to my companion, Stefan Compant, who is leading the scientific coordination of the project, together also, of course, with our hosts here, CNR, the National Research Institute of Italy, and CRSFA, and they're well represented here. So we're great to have everybody here. And Stefan, I hand over to you. Thank you.
Thanks, Stephen. I think you said all of the things I wanted to say too. Just welcome everybody, and you will see what we have done since 5 years in BIOVEXO. We put a lot of effort with academia, RTO, companies during a 5 years project granted by CBGU, previously BBIGU, and you will see some of the results we got after 5 years. And I remember 4 years ago we were there, just after the COVID, as you see, as you said, Stephen, and we were starting to develop the project, and now we are already at the end. It was 5 years, but it's a short time, you know, to develop some solution according to the situation of Xylella in Puglia, and also I'm thinking situation of Xylella in Mallorca or Spain, in the mainland. And I'm very happy to be there, and I'm sure we'll interact a lot with question, answer during all this day. Thank you very much. And the next person.
So, Franco. Oh, so, and okay. That's fine. Whoops.
Okay. Good morning to everyone. It is a pleasure to welcome you all to this final meeting of the European BIOVEXO project here in Locorotondo. I bring the greetings of my institute, CSF Basile Caramia, as well as those of our president, Michele Lisi, and our director, Professor Franco Nitro, who unfortunately could not be present today to unpostponable commitments. Having been personally involved in this project, and I am particularly pleased to share with you the result of this project. BIOVEXO is an European project that has brought together with 17 partners, including scientific institutions, industries developing phytosanitary products, and farmers, all united by the common goal of developing sustainable and effective solutions against Xylella. After an initial screening phase of the selected organic and innovative products, various field trials were carried out, first on a small field trial and later on large-scale field to evaluate the effectiveness of microorganisms and natural substances in control of the bacterium Xylella fastidiosa, as well as in integrated management strategy to control its main vector, Phyllogenus fumarius. This activity have demonstrated our collaboration between research, industry, and agriculture can generate a promising preliminary result for the control of this devastating disease. On behalf of Professor Nigro, I would like to thank you all partners for their excellent work and cooperation through the project. I also wish to express my personal gratitude to everyone, but in particular to my collaborators for their efforts and the work carried out to develop the project activity. Greetings to all BIOVEXO partners for their professionalism and passion in making the project. Thank you all, and I wish you a productive and inspiring meeting. Thank you.
A final introductory word then from Francesco.
Yeah. Thank you very much. My name is Francesco Di Serio, and as a director of the Institute for Sustainable Plant Protection of the CNR, it's a real pleasure for me to welcome you here. As you know, our institute is a reference institute for plant disease studies in Italy. The institute has several research units distributed in Italy, and Bari is one of these units. And of course, we work on Xylella, but the institute has a wide approach to plant disease, so it's a multidisciplinary orientation. So we also work on entomology and virology, and all the plant disease are covered, and also abiotic stresses. But of course, Xylella is one of the main topics. As an institute, we are making big efforts to address this really important and devastating pathogen. And I'm really happy to be here to also to know more about this project. But as you know, this is one of the most important example of how also our institutions, but all the partners involved, may do research that is not closed in the laboratory. So this project is a clear evidence that research can go in the field, can test the results, and events like this of communication is really important to communicate what has been done, what are the future perspectives in this direction. So I'm really proud to be part as an institute of this event, but of the project in se, and to contribute to these big efforts to reach the field, to solve the problem with an ecologically sustainable approach, and with so methodologies that may really fulfill our inspiration, that is to make sustainable plant protection. So thank you very much, and good work to everybody.
Thank you so much. So that was our introductory remarks. I will invite you then to take a seat back in the audience. We'll get started. Maybe just to get a rough feel for who is in the room, right? Who's on the side of more like research, right? Public or private, academia, research institutes. Who's working in this kind of area? Okay. Okay. Who's more on the, say, business side, product development? Okay. Public authorities, maybe local or national, European, a couple of public authorities. Anyone sort of civil society style, NGO maybe? Okay, students? Students? Couple? Yeah, half students. PhD style students, okay. All right, okay, so more on the research side, so that's useful to know. Okay, so let's get started. We'll first then hear from Dr. Karin Nienstedt. She's Policy Officer at DG Sante at the European Commission, and she'll be giving us an overview of the regulation around biopesticides, but then also with regard to Xylella fastidiosa. So please give a warm hand to Dr. Karin Nienstedt.
So can you hear me now? Okay. Buongiorno signore, buongiorno signori. I will continue in English. I think that's better. Thank you very much for the invitation and also for the introduction. It's a pleasure to be here in Locorotondo for the final conference on BIOVEXO. I am Karin Nienstedt. I work for the European Commission in the unit of plant health. That's in the Directorate General of Health and Food Safety. I joined recently the unit. It's not working. I can move it. So you hear me better now? This way? It's okay? So I joined recently the unit, so Xylella was, of course, not completely new for me. I worked in other areas of crop protection before, so it's a particular pleasure to be here on BIOVEXO to learn also more about your results. I will give you a regulatory update, so probably there's not a lot of new things for you. On plant health and what concerns Xylella, and also as regards regulation of pesticides or biopesticides. What are the steps to be done? So next slide, please. Oh, it's me moving it? Okay. So as regards the legislation on plant health, the legislation currently in place, it's, let's say, the big regulation on the plant health law, on protective measures against pests of plants. It's a relatively new regulation. It's from 2016, so after Xylella had the first outbreaks. And then as regards Xylella fastidiosa, is this working? Can you click maybe? There are a series of, let's say, daughter legislations. Can you click again? It's not working. It's not working. No. So there are a series of daughter legislations, implementing regulations, delegated regulations. One which is implementing regulation 2019-2072, which is the big implementing act to implement the plant health law, which has the list of quarantine pests. Of course, Xylella fastidiosa is listed as quarantine pest. Then you have a delegated regulation listing priority pests, which also lists Xylella fastidiosa. And then there is an implementing regulation from 2020, number 1201, which specifies the measures which have to be taken to prevent the introduction and spread of Xylella fastidiosa in the EU. I think that's not new for you. As regards where we have Xylella at the moment, this map is really new. It will be published in a few days on the Santé website, and it has been updated recently to show this little outbreak in Gargano in Italy, and also this big area in France, which was changed very recently from eradication to containment. So you see Xylella is, I mean, if you compare it with a map of 4 years ago, it's moving and it's advancing. So as regards the history on Xylella, I mean, this is a bit of a timeline. So as you know it better than me, there was a first outbreak or detection in 2013. And then there was a series of, let's say, EU legislations to try to control the situation in 2014, in 2015, based on information prepared by EFSA here. And then, so you see, EFSA has been working continuously since 2013 on the topic with several updates on the scientific information. And then in 2020, there was the implementing regulation. This was, I mean, all the decisions were superseded by the implementing regulation 2020-1201 because of the change of plant health law, which was here in 2016. So this implementing regulation was then amended a few times as regards the host plant species and as regards the containment zones. The last amendment was just voted in September a few weeks ago, and it was published a few days ago, actually, last week, I think. So what is important to know is that last year we have asked EFSA to update the scientific risk assessment, and once we have a new compilation, let's say, of the last state of the art, then we will see what needs to be done and how we can update the, let's say, regulatory situation at the moment. But at the moment, we are waiting to EFSA to see what is the update of the scientific information. And I think Giuseppe will probably give us more information on this. So, just in a nutshell, the last changes in the implementing regulation are the definition of the vector of Xylella, that it clarifies that when a pest is found in the vector, when Xylella is found in a vector, the surface needs to be identified in the infected plants to establish the demarcated areas. It updated the list of high-risk plants, and since 2024, it made a little change for the containment zones in a way that it changed the zone between the buffer zone and the infected zone where the surveys have to be identified. So this was reduced from 5 kilometers to 2 kilometers. So that's on Xylella. I would like now to move to the regulation of biopesticides and what are the steps to bring them to the market. I understood, as it was said, that BIOVEXO is about finding solutions. That's fantastic. I am looking forward to the day to see what are the solutions, but to be able to use this in the farm, they need to be, let's say, put on the market. There are a series of regulatory steps which have to be done. And these falls under the regulation of placing on the market of plant protection products, which works in a nutshell like a pharmacy. It's putting solutions to the farmers on a shelf. It's not regulating the use, it's making solutions available. So you could compare it like a pharmacy where you go to buy medicines or to a farmer's toolbox so that the farmer can choose the tool which they need for solving the problem. So what is a plant protection product? As a reminder, it's a product, usually liquid. It could be also have other formulations, which contains an active substance. The active substance can be a chemical or a microorganism under the legislation, and it may contain other ingredients like coformulants, safeners, or synergists. That depends on the product. It's working as a two-step authorization system. That's important because there is an approval of active substances at EU level, and there is an authorization of the plant protection product uses at member state level. So there are two steps to be done. As regards the active substance, it works like a positive list of active substances. Same applies to safeners and synergists. So the substances need to be approved and put on a list, and only then they can be used in plant protection products. This means that the burden of proof is for the applicants. No data, no market. That's the principle. So the applicants need to prepare a dossier which contains studies, or let's say data requirements, which are defined by law, which have to be done via GLP, good laboratory practice, and the dossier also needs to include peer review literature. Depending on the substance, they may be categorized or regulated as different kinds of substances, low risk, candidates for substitution, basic substance, or let's say the normal or regular ones. And there is a periodic reassessment. So it's not an approval forever, it's an approval for a definitive time. Then as regards coformulants, that's important, it's a negative list. So coformulants are also regulated, but there is a list of unacceptable coformulants, and there is a process to add, let's say, substances to this list of unacceptable coformulants, but there is not a positive list like for the active substances. So the first step of approval means that an applicant needs to prepare a dossier. Then this dossier contains the data requirements under first risk assessment. Then this goes to member states, to a rapporteur member states who makes an assessment. Then there is a peer review by member states and EFSA, which ends with an EFSA conclusion. And based on this EFSA conclusion, then the Commission and the member state decide if the substance can be approved and put on the positive list or not. It's a clear separation of risk assessment and risk management. There are a lot of scientific experts involved. It's a process which takes 2.5 to 3 years, and often longer because of several reasons. And the approval is for a maximum of 15 years. Then it would need to be renewed. Then, if the substance is on this positive list, then the second step kicks in for the authorization of plant protection product. And this means that each member state could authorize a plant protection product. However, in order to make it easier, Europe is divided in zones. And then, let's say there is a common assessment done in each zone where there is an interest to authorize the products. And member states cooperate. And then once there is an authorization of our plant protection products, other member states could mutually recognize. There are other provisions. Member states could authorize minor use extensions. These are cases where industries may not be interested to submit a dossier for an authorization, but there is already a product on the market which could be used, and then member states could extend to minor uses. And member states can grant emergency authorizations. These are they are fast, they are quicker. They can be granted by member states for problems where there are no solutions available. So the good thing of emergency authorizations is that they are fast and pragmatic, but they are only for 120 days. Of course, if there is a need, they could be repeated, but they are limited in time. So for a normal authorization, it's 16 to 22 months. Also here, there are many delays. And the duration of the authorization is the approval of the active substance plus 1 year. So for an applicant who wants to get a new active substance or a new plant protection for the market, what should they do? So they would need to prepare to make an application for approval of an active substance. That means they need to prepare a good dossier and get ready early in time. They need to think what are the representative plant protection product uses they will put in the dossier. If it's one use, if there are more uses, I mean, these may be strategic decisions to be taken. They need to think which data are needed and how to organize the submission via Euclid. Euclid is an IT system. It's quite big. So there may, some advice may be needed there. So it may be a possibility to get advice from a consultancy or to talk, let's say, to people. Then for new active substance, you can select the rapporteur member state. But it's important to get early in contact with rapporteur member states because sometimes there are waiting times for the different member states. So it's not that an applicant comes and say, I have a dossier and here I submit it and it's done. Sometimes they say, okay, but I can only look at it in 2 years' time. So you need to get early in contact with rapporteur member states. And then there is the possibility to ask for pre-submission advice in member states and in EFSA. So that's to clarify the scope of the dossier, maybe, or what is needed. And that's also a possibility which is good and which could be planned early. Then once the dossier for the approval of the active substance is submitted, it's good to follow it up in order to get ready in time for the applications for the plant protection product in member states. So there are quite a number of implementing regulations which are relevant. The list of active substances, which is this one. Data requirements, we have 2 implementing regulations, one for active substances and one for chemicals. One for active substance, one for plant protection products, and we have uniform principles of evaluation. They are relevant. For each of these, there are always a part for chemicals and a part for microorganisms. Important for the microorganisms, there were quite recent updates to make it easier for microorganisms to access the market. Then for coformulants, the list of unacceptable coformulants and the labeling requirements, which may be also important for the dossier compilation. For biopesticides, in particular important, the need-to-know approach, which is it's implicit in the legislation. For microorganisms, I mentioned the recent update. With this update, the biological properties were put at the center, and this means that some data requirements are conditional, depending on the particularities of the biology of the active substance. So that means it's really need to know and not nice to know, and there is a scope of conditionality in the data requirements. For the part A on chemicals, this also applies to some chemicals which would be regarded as biopesticides, like pheromones, botanicals, peptides, antibodies. So this falls under the chemical part. However, also here there is a need to know implicit, which if I put this on the screen because I think it's important. The data requirements for chemicals are quite extensive, but there is this general point in the introduction of the data requirement which says that some data may not be needed under certain conditions, which are basically scientific justifications that it's not needed because of the nature of the product or the uses. So there's always the possibility to waive data based on these provision. So need to know approach, and there is a problem formulation guidance document which could help to find these justifications. Guidance documents are in a database which you could find on Santé, including this problem formulation document. And that's the situation today. We are working to make it easier for biocontrol plant protection products to access the market. And there are two things I would like to announce. This is not yet available, but it's in the making, so keep an eye on it. So one is that there is an amendment for the data requirements ongoing, where the public consultation closed in October. Possibly it will be voted at the standing committee in December, and then there are 3 months scrutiny of the Parliament and the Council. If these don't object, then the act could be adopted more or less in the second quarter of next year. And what is important in the context of biocontrol is that this part 1.5, which I mentioned in the previous slide, we have made it more explicit, mentioning microorganisms, semiochemicals, or other substances originated from or secreted by living organisms, that they would be applicable to get, let's say, this derogation. And we hope that these would facilitate the justification process. And then another thing which is in the making is, and that you may also have heard about, is that we are preparing a food and feed simplification package, which will contain some targeted amendments to the plant protection product regulation, which includes a definition of biocontrol active substances, the possibility of member states to grant provisional authorizations for plant protection products containing such new active substances before the approval process is finished. So while the evaluation is ongoing, these provisional authorizations could be granted. Then there would be a fast-track procedure for the approval and authorization, and additional resources to EFSA to speed up the risk assessments. So there was a call of evidence on these, which closed in October, where member states and stakeholders could provide comments which were considered in the draft. So the draft is, of course, not yet public because we are working on it. Once the proposal is adopted by the Commission, then there will be a negotiation with Parliament and Council. So this could take some time. So on this, let's say, process, I cannot give you more indications as regards the timing. So that's it from my side. I put a slide with, let's say, important links, which may be useful. And that's it. Thank you very much for your attention.
Thank you so much, Karen. Maybe a couple of questions, if you'll allow, and also feel free to raise your hand. I have a couple, but okay, we have one here. I'll start with you then.
Pasquale Saldarelli from the CNR. Several questions. I learned in BIOVEXO the biostimulants and plant protection products. So this distinction I need to know. Another question is, you say that if a product is present on the market, and even an NGO, farmer organization, can promote, can ask for its use as a plant protection. Is that correct?
Okay, thanks. Thank you for the questions. Indeed, the issue with biostimulants, that's something which still is a little bit under discussion, because in the fertilizer legislation they put biostimulants, and there are some products which are a little bit like, it has to be clarified if they fall on one side or another. There is an ongoing discussion on these, also with member states, to clarify these. If you have a particular question, I would advise you to go in contact with your competent authority, your national competent authority, to clarify these, or to come back to us with a particular question so that we can look at it. As regards the issue of authorization of plant protection products by other, let's say, parties which are not the applicant, what I meant is that under the minor uses and the mutual recognition, processes. If the industry is not submitting, let's say, an application for an authorization of a plant protection product, there is a provision in the legislation that third parties, academia or national institutes, could do so under certain conditions. The thing is that it has not been used very widely so far by member states because there are also fees associated with these dossiers and the applications, but there is a possibility to do that. So I can only advise also here to get in contact, if you have such a case and you have an interest, to get in contact with the local authorities to see what can be done. And this applies for mutual recognition. It's not for new authorizations. It's for authorizations which are already, which exist and which then would need to be, let's say, for mutual recognitions, if it exists in another member state and you would like to get it authorized in your member state. So that's the mutual recognition. And for minor users, there is an authorization in your member state for a particular crop, and you want to expand it to other users. So there needs to be an authorization in place, but then there is the process that these third parties could ask for either mutual recognition or minor use extension, indeed, yes.
And on the authorization, so you mentioned, up to 2 years, potentially with delays, or it's a long process. Out of curiosity, if there's a product emanating from a European-funded project, could it arguably benefit from some kind of fast-track process because it was, emanating from, let's say, taxpayers' money and an investment from the public authority, or would it be treated completely equally as any other applicant?
I think that depends on the member state. That's why I said it's important to get in contact very early with the rapporteur member state you are thinking of. I know informally that some member states, they are very pragmatic. They say first comes, first serve. So then there is a new application and it goes at the end. So, but there may be other rapporteur member states who have a particular interest who would be maybe ready to give priority to that. So it depends really member state by member state. And that's why it's important to get in contact early to clarify this. Also, if it's a new active substance, there may be, there is the free choice of the rapporteur member state. There may be rapporteur member states who are more interested than others to assess a certain dossier. But also that needs to be, there is a need to get to the competent authority and to say, I am preparing this, would you be interested to be a rapporteur member state? And let's say to see what is the situation. So early contact is important.
Okay. And maybe last one on, so you talked about the regulatory framework around plant protection products. Has there been any assessment done on the interaction between the spread of Xylella fastidiosa and the European regulatory framework on plant protection products, i.e., has the regulatory framework had an effect on the spread of the disease because maybe, I don't know, it prevented a certain substance of being used or something like that? Has that kind of assessment been made? Or would you argue that the spread of Xylella fastidiosa has nothing to do, let's say, with the regulatory framework in the European Union?
Well, to my knowledge, I am not aware of such a study, and probably this would be then also an issue to be studied in the member states concerned, because as Commission, we don't have the overview of which are the plant protection products authorized in each member state. So that's a national issue. So that's more for each of the member states to look at. Well, what I know is that Xylella is biologically a very complex and difficult disease to tackle because there is a vector involved, so it's not such a straightforward thing. And it's something new, and so science needs to develop solution, and then this needs to come to the market. That is not happening in 1 year, in 1 day, unfortunately, also not in 1 year. Maybe just back to your previous question on the delays. Very often the delays are also caused because the dossiers may not be complete, because maybe the applicant thought this is not needed, and then the member state says, yes, but I need this. So, and that's again, back to my plea, get in contact with the rapporteur member state and EFSA with the pre-submission advice early so that you can really prepare a good dossier, because if the dossier is good, the process runs. If there are follow-up questions, then the delays occur because there are months between the authority coming back to the applicant coming back with the answer, maybe generating new data, etc. That takes a long time.
Okay, thank you so much, Karen, for your time, and you'll be with us all day, so if you have any questions for Karen, I'm sure she'll be able to take them throughout the day. Please give a warm hand then to Karen. Thank you. So Karen also mentioned the importance of the scientific opinions provided by EFSA, the European Food Safety Authority, and that then informs policy, but it also is a very important organization in terms of authorization. So now we'll be hearing from Giuseppe Stancanelli, who's in charge of plant health risk assessment at EFSA. Please welcome Giuseppe.
Thank you very much. Can you hear me well? Yes. So thanks a lot to BIOVEXO for inviting me to this conference, and I'm very, very keen and looking at the results of this European funded project for the control of Xylella and for biopesticide. So my presentation is a follow-up of one of the two topics were presented before by Karin Nisted. Is about risk assessment, plant health risk assessment supporting the control of Xylella fastidiosa and the link to research. And I will not talk about pesticide authorization, which is also done by EFSA, but not by my team today. So these are the four topics I would like to briefly present you. So what are we doing to update the risk assessment of Xylella in these years? So we intend to finish by end of next year. Something about the EFSA-funded targeted focused research on knowledge gaps on Xylella, which is different from the European project, it's much smaller. The European Conference on Xylella and the communication campaign Plant Health for Life, very briefly. So we received from DG Santé a mandate. DG Santé tasked us in 2024 to update the 2019 risk assessment on Xylella, which was quite recent, was 5 years before concluded. The reason for this new mandate is to look at the specificity of the European oryx and the different subspecies present in Europe. So situation in Europe has evolved, we have new oryx, different subspecies present. So we will look at what we've done in 2029 for Xylella in general, Xylella fastidiosa in general, and focus on the data we have from the European oryx and on the different species. So we look at spread impact, scenario for eradication or containment, risk reduction option. At the end of this process, in the course of 2026, we will open our risk assessment to a public consultation where we ask the public, the citizen, the scientists to contribute by commenting and sharing evidence. So we're not asking to say we like or we don't like what you are saying, because that's not very useful for us. We will ask, look at what we have done. You think we've done right or wrong? Do you have other evidence that we didn't consider? So that's the purpose of a public consultation. After that, we revise the risk assessment and finish by end 2026. A lot of people involved in this, so we have a working group of experts, panel members, scientific experts, more than 10, from microbiologists, plant pathologists, modelers, entomologists, etc. We have my unit, EFSA Plants unit, with many colleagues, methodological unit in EFSA, but we also have collaboration with research institutions through grants. We work with the CREA in Bologna, with the CNR in Bari, with University of Bari. We have external consultant, and then we also consult with specific scientist expert or national expert from phytosanitary authority for particular information, data, evidence. They are called hearing expert. That's a small photo of only the people that were there when we took the photo in that meeting, but the group is much bigger. As you see from this plan, you can read it, probably very small, but the plan is to conclude most of the activity in Q1 and Q2 2026. So let's say before the summer to conclude most activity, and to launch the public consultation. We would like to launch consultation on more technical annexes between Q1 and Q2 2026, and the consultation of the full assessment will be launched in the summer 2026. And here I present you briefly different activity we are conducting. I cannot show you the final results because the work is still ongoing, but I will give you some hint of what we are doing. So we work on the establishment for different subspecies. We look at the climate suitability, the host range, the host distribution, the vectors. We get data from the member state. We have a very good contribution from all the member states, from Spain, Italy, Portugal, and France, where we have the BRICS to get the data. We get continually update of the data of what is the current situation, and we are getting our new data. We look at the data we have collected in years in the host plant database of Xylella. We reference the position of the different plant found infected. We look at scientific paper and additional sources. So by this work, we'll be able to see exactly in which area, which climate, Xylella, different subspecies are present, and then we try to model, this was already done in 2019, now we do an update, we try to model to see which area of Europe is more suitable for the different subspecies. We also will look at the species, so if we do the model for the climate, we also have to put in a map the different crops at risk, so we look to different sources, the different crops and vegetation by different subspecies which can be affected. And then we look at the vectors. For vectors here we have a specific grant, a cooperation grant with the University of Bari, with Cortinelli Daniele Cornara, where will help us in collecting from the literature all the data on the updated data on biology, ecology, the different vector of Xylella, and focusing first on olive and grapevine, which are the most concerned at the moment in Europe, and then on almonds and the other crops. So then we also looked in 2019 with the incubation period. The researcher, the scientists do a lot of experiments by inoculation, and this allows us to see how much time does it take in certain conditions to develop the symptom. And this is an update of the previous opinion, 2019. We look for data on both the latent period and the incubation period. We also did a call for data, so we launched on the website a call for researchers to share with us even unpublished data, and now this is part of our evaluation. And we're looking at a certain number of studies. In fact, at the end, out of all the search, we found 43 studies which on incubation period and 14 on latency period, which can allow us to extract data, because sometimes a published paper is very useful, but maybe doesn't contain enough data to be analyzed. So, and we are now doing the statistical analysis of this work, which is ongoing. For the spread, we try to use mathematical models to look at the spread and to look for the short and long jump spread. So not only for the spreading around one infected orchard, but also how sometimes this disease with several means can jump to a different distance. And this work is done mostly with our colleague, Martina Sendoia from IVIA and Rachel Russell from Cambridge University. And we cannot model everything. I mean, risk assessment like science need to focus on particular aspect because we cannot do everything. If not, we still would keep doing things but never finishing. So we'll focus, of course, on the olive infection with Xylella fastidiosa subspecies pauca in Apulia. We'll focus on the almond infection in Alicante with the subspecies multiplex. We'll focus on Xylella fastidiosa. The data we received to start the activity was mostly from Mallorca, where they have fastidiosa subspecies on both grape and almond. Now we have new brick also in Apulia from these subspecies. And then we will focus on Spain, on the data we receive from France on non-crops, so natural vegetation in the south of France. You've seen it before in the slide by Karen Nisted. There is this blue area in the south of France, where Xylella is present and under containment, but is in natural vegetation. About impact, this is the final part, the final most important part of the assessment will be done in the course of beginning of 2026. We will work a specific host, a specific subspecies. Much was already done by EFSA with many different experts in the recent years for the work on priority pests. We already have some work done for around 50 pests, including Xylella. But here we'll focus very much on the impact of a particular subspecies in particular crops. And we're now preparing to do this work. For risk-reducing option, we are working on 4 levels. We are working on the vector control. It's very important to control the vector of a disease which is transmitted, in this case, by a native insect. We work, on substance which can control the bacteria in the plants, and here maybe we are very, very near to the presentation of today of BIOVEXO, on agronomic practice, and then on the tool that we also have for controlling new pests, which is use-resistant or tolerant plant variety. Vector control, in summary, this work is almost finished, and we had enough studies available for vector control. We extracted data at the end from 72 papers, and we can do a statistical analysis, meta-analysis of the results. And particularly, we look at the difference between the survival of the insect in the control respect to the treatment. We can classify the paper based on different level of possible bias in methodology, no bias, minor bias, major bias, or paper which are excluded because either not relevant or they really cannot be used for the analysis. Because the asset, for example, there are some paper where there is no control, so you cannot really make a comparison. And this will be shown then in a graphical way at the end. What we can say is that there are best treatment which reduce the efficacy, the survival of the insect vector up to 80%. Generally, these are authorized pesticides. Some are non-synthetic pesticides. It is important to see that there is also efficacy of biocontrol made by fungi or nematode, which is significant, however, is lower at the moment than the synthetic pesticide. The use of predator is highly uncertain, probably because we deal with a native insect, so it's where everything is already established in the environment. And we are collecting data worldwide from all the literature on all the different species of insect vectors. For implant control agronomy practice, we are looking at all the paper published. We will keep this open until the moment we finalize the assessment, until next year. However, we have a less number of papers. There's much less published on implant control and also in agronomic practice. We're talking about 20, maybe we're right to 30 paper maximum. So yeah, we cannot do a statistical analysis, we'll do only a narrative description of the results of this paper. And for resistant variety, we have also a grant, a cooperation grant with CNR from Bari, coordinated by Maria Saponari, where the colleague from CNR Bari will help us in reviewing the resistance tolerance varieties, starting from our host plant database and looking at literature, and will also help us in providing an update on taxonomy, on phylogenetic analysis, date of introduction, and impact of the different MLST type of Xylella. I don't put the name of all the collaborators. These grants will be all our knowledge in the end, but now I only put the coordinator for a reason of brief. So what we do for public consultation, so incubation period and potentially also the control of vector should be ready soon. So in the first quarter of 2026, we will launch a public consultation. For the other risk reduction option, the second quarter of 2026, And the full opinion of the public consultation is targeted to be launched during the summer 2026, so that end of 2026 we have a deadline by the European Commission on November 2026. We target to finish the work. So then the other topic of today is why we EFSA fund research project on Xylella. So EFSA fund some particular target research project when we, doing the risk assessment, we identify area where there is a lack of published evidence that is really needed for our risk assessment. So we have done this for Xylella, given the importance of this pest for the European agriculture and environment in several instances. And in green you see the one which are concluded, in blue color the one which is ongoing or has to started. So the first one was the pilot project with CNR Bari on the host plants of Xylella fastidiosa, and I was very, very happy to hear from Maria that now the field we initially funded, but then was continued by the Ponte Bexil project, is now in production for some resistant variety are now in production. Then there was a study with different Italian institutions, university, Brescia, Torino, Bari, CNR from different institutes working on the biology, collection of data of biology of the vectors in Italy. Then we have a study in Balearic Island, and a scientific study collecting the data on the biology of the vessel in Balearic Island. But the scientists there, they gave us the idea that we should communicate the results also to the citizens, to the children, to the general public. And with this idea, we make a small comic stripe cartoon that show to a bit what happened on Xylella Europe, which is published on our website and is used by several media. And now ongoing, there was a very worrying outbreak of an invasive potential vector Xylella in the area between Spain and France recently, 2022. Sharpshooter, Draculocephala robinsoni, luckily is not a common pest in agricultural crops, it's mostly in the environment, but we funded a target study to look at the biology of the transmission of Xylella by this insect, which involved Spanish institutions like SIC, IRTA, University of Bari, and Foundation Edmund Mack in Italy from Trento. We have currently a project in US with University of Leuven in Belgium. They work with Virginia Tech and UC Berkeley. Why we have this project? Because we want that it is studied in America what happened with Xylella in the cold area on the temperate crops. So at the moment, Xylella is very concentrated in Europe, in the Mediterranean area. So we have outbreak in Spain, in France, in Portugal, in Italy. We don't know exactly what will happen with Xylella when it arrives to Central or Northern Europe. And we have the possibility to study this, what happened in US. US, Xylella is a common pest. It is not an area of big research. They don't fund much research. So we have a project which looks particularly in grapevine, in the northern area of distribution of the grapevine in US, what happened with Xylella, and in forest, and particularly focus on Xylella multiplex, which is the subspecies which, based on the climatological modeling, seems more suitable for the northern climate in the EU. And the last things, we just launched in 2025 a call for proposal for doing research on the epidemiology, the biology of the vectors in table grapes in Apulia. Is not yet signed the contract, but the awarded project should start to work in early January 2026. This was for us a very important problem to see the table grapes as an outbreak of Xylella transmitted by vectors. So we try to focus research to understand the epidemiology, so this can be helpful for the farmer for the local authority to control the disease. And each of these projects need to communicate and disseminate the results. That's very important. We do this together with scientists, stakeholders, in the scientific conference on Xylella, the European conference, that is run every 2 to 3 years. We did 4 so far. One was online during COVID, the other in Ajaccio, in Balearic Island, in Lione. And the next one, I'm happy to announce, will be here in Puglia in Bari, the last week of June, 22-26 June 2026, and we'll organize together with the local authorities, the Plant Protection Service, and the European Research Project on Xylella. So communication is key. This is a campaign by EFSA. There's also a campaign by EPO at the airport to explain to the passenger the importance of plant health, the risk of plant health. The message is don't bring plants. You can bring pests with the plants. And it's a campaign we now run for 3 years. All the European members, the neighboring country are involved. So it shows that it's important that we talk with the people. And to conclude, so to do risk assessment, we utilize all your research results. And by conducting and publishing research results, you can provide evidence for the risk assessment, and particularly also very much more important for designing solution for the pest management. So communication and interaction with stakeholders is essential for risk assessment and for research. And so we invite all the scientists to keep sharing and publishing the results, to participate to EFSA consultation, and to join the European conference. So thank you.
Thank you very much, Giuseppe. Quick question for you, because you talked about the importance of communication, right? So we see that there are projects like BIOVEXO that are trying to find solutions to a problem. On the other hand, we're still also trying to understand the problem, right? I mean, that these two things are going on in parallel. And you're doing a lot of research, let's say, looking at academic papers and trying to understand the science around this. I'm just wondering, how much are you engaging also with, let's say, growers on, let's say, real field experience of the problem, and how much are you able to feed that in also to your research on understanding the problem? Because that might also help in the end to communicate results, right? If growers have been somehow part of, sharing their experience, and how much that can help you better understand also the disease, even if it's not necessarily a scientific approach in that sense, but it's still grounded in very real experience in the field.
Thank you. That's a good question. So EFSA is based in Parma, serves all the European Union. So we focus on evidence and data. The evidence and data is produced by the local researchers and is collected also by the local authorities. So we don't have the capacity to go around and meet all the farmers, even we would like to do it. So what we try to do it for our work is we try to keep our fit into reality. That means that our working group is traveling to the outbreak area where we're in outbreak, is seen, accompanied by the local scientists, by the local authorities. We visit the outbreaks, we try to understand in practice how it works. We interact with the local stakeholders, and particularly the authority and the scientists, to get feedback on our results. We try to communicate. So we try to serve the farmers, although we don't have the capacity to meet and discuss with the farmer. I think this is more the task of the people working in the place. We cannot think that we can do everything. But I think it's a key. It's a key. The key message is what we do should be useful to the farmers, to the growers, to protect their crops at the end, because even the European measures are done to protect the crops. So the measures done to reduce the spread of Xylella are protecting all the crops outside the containment area, and the measures designed by the local authority in the containment area are meant to support the farmers in living with this situation, which we know is very difficult.
Okay, thank you. Do we have a question for Giuseppe? Or a comment, a reaction? No? Okay. Thank you so much.
It's always a very, very nice message that there is no question, and very clear.
Very clear.
Or maybe it was very unclear. Nobody dared to ask any question.
Thank you so much. Thank you. So we've heard on some of the research, the context, the regulatory context, but also the research on risk assessment. Now we're going to go a little bit deeper on some of the responses, and we'll start first with the area here in Puglia, the local area, Italy and Puglia. So I would like to invite Dr. Donato Oscia, Research Director at the National Research Council of Italy, to tell us a little bit about the Xylella scenario in Europe and Italy. Please, over to you, Donato.
Okay, thank you very much for this presentation. I want to say that I'm former the research director, because now the retirement has to. Okay, in any case, let's start with my presentation. I will try to give a kind of a short overview of the situation, as you said in the presentation, of this story that, as all of you may remember, started 12 years ago in this area, in this region, with when the olive quick decline syndrome started to show up. At that time, interesting, looking now limited part of the territory, but at that time it was already a quite large area. If you want to try to eradicate one organism that is also carried by flying vectors. This is okay, this map is already obsolete because we learned by Karin this morning that there is a new version. However, even with this that is not updated, you can see as surprisingly from what we knew 12 years ago, the presence of situation of Xylella in the European continent, I will say, or in the Mediterranean area, is not so rare, because, as you can see, there are a lot of outbreaks, so a lot of introduction that occurred in the time. And I underlined a lot of different introduction, because it is clear that it's not the spread of the same strain, but because you can see that already have been detected 11 out of the 90 sequence type that are known for Xylella fastidiosa. So, but this is has been the consequence of 11 years of mandatory monitoring program, because the monitoring program became mandatory for each member states with the the Commission decision 789 of 2015, and now it is quite interesting to learn by Karin that now in the French mainland they switched from the eradication to the containment, and but you can see now Xylella has been identified also in Israel and in Lebanon. But the important point to keep in mind is that while Pauca 12 years ago was discovered as consequence of the phytosanitary investigation that were implemented at that time, because there was a very impacting problem going on the territory, in most of the other cases, the finding occurred just only simply as consequence of the monitoring program. Not because, except few cases, but in most of the cases, there were not impacting diseases observed and reported by farmer, by a scientist, by a plant protection service, but doing the monitoring program, these were found. And this is true, of course, mostly for the other two subspecies. First of all, the multiplex, and you can see the multiplex is the most spread, the most present in Europe. Oops, what? Sorry. Okay, so this is a map of 2000 of the pest risk assessment of EFSA of 2019. Now we learned by Giuseppe that probably we have to to modify a little bit the meaning of the climatic suitability, because according to what has been discovered in the States, in Virginia, I think, because Giuseppe just said about this. But however, at this time we can consider that almost all the outbreaks that are present in the map of the European Commission, they fall in an area with high or moderate climatic suitability. So there is a quite good overlapping with the map of the climatic suitability. And therefore the climate, we can consider the climate as a key factor for the epidemiology of Xylella fastidiosa, together of course with the vector population and with the presence of just a curiosity, just to give credit to Professor Alexander Purcell, that already in 1988, with empirical tools, because now the modelists of EFSA, they have a lot of very sophisticated tools for doing this work. But and you can see that map is a handmade map that in 1888, at least for the case of Apulia, show in 1988 already a situation of high risk that is more or less overlapping with the real situation today in Apulia for the case of the olives. But also the climatic suitability, it is different according also to the subspecies. And as Giuseppe said, the multiplex is the one that that can be also spread, can spread also in area where the climate is more cold, while Pauca is more warm-allowing than multiplex. We do not have to forget also that we have to take in account the climate change. It means that probably, beside the other factors, but just only for this factor, probably the maps of 1980 of the 2019 probably are not actual anymore. But this is okay. Is this one that the previous slide was a old one that by mistake remained in the presentation? But this is more or less the current situation in Apulia. The colleague that will follow my presentation from the Plant Protection Service, that by the way, in the program is written Salvatore Infantino, but it will be substituted by Dr. Francesco Palmisano from the same office. He will give more detail on this. But I named the Apulia a good case study for this in general, because so far in 12 years are already over 1,400,000 plants that have been analyzed in laboratory. I think, just to give you, I think that the magnitude of this program is unique. It's not, I do not have information about similar, so big, so enormous campaign of monitoring done in other parts of Europe, but of the world at the same time. And then I think that even if not sustainable for long term, this kind of campaign, but can give a good contribution, an important contribution for the real understanding of the real situation of Xylella in our regions. And just a few words to remember to the platea that which has been the impact of the Pauca on the olive industry in Italy. There are several millions the trees, olive trees, that are severely compromised already in these years. And by the economic point of view, there are just this year was published one study of economists of the University of Bari that using real data, they made a first estimation of the damage occurred so far, and they estimate in a total loss of 132 million euro per year of profitability, and a total loss of over 1 million working day per year. But there is, it could be also a kind of underestimation, because the database that they used to do this was not so, probably was not large enough to estimate. But in addition to this, there are other damage that should be taken in consideration. One important is that the big loss of the value of the lands now in that area, as well as the environmental and climatic damage that was caused by this epidemic. And sorry, what happened? So the limited spread in the northern outbreak of Pauca is not. So now we saw also by the slide of Karen that now there are in Apulia, in north and central part of the region, are there several outbreaks of Pauca. However, the spread of those outbreaks is not comparable with the devastating spread occurred in Salento. So now it is true that there are several outbreaks that are appearing in the central northern part of Apulia. But in addition, we also have to say that at least a part of this spread are a consequence now of the spontaneous report made by citizens of suspicious cases, because probably there is an increase of sensitivity of the people in northern part of Apulia that started a kind of spontaneous case of citizen science, because they are reporting these cases. They are helping to discover also outbreak that in some cases probably they are also old outbreak, but small outbreak, because the spread seems to be much slower than what occurred in southern part of Salento. What happened? Just to remember that on the left you can see which was the situation 12 years ago, but in 5 years there was we observed a very fast spread of the bacterium, and that in the center is the official demarcation made by the European Commission in 2008. And if you look in the case of the area that is under containment, the further expansion of the infected area since 2018 has been very limited. I can put that yellow oval on the map on the right, and this is the situation. So the spread, but even the spread over short distances, in the case of new outbreak in the north, is much slower than it was in the south. Just to give you one example that I remember by personal experience, that 12 years ago, when in one olive orchards in the southern part near Gallipoli started to show some symptoms, within a few months, the whole orchard was interested by the disease. While in the central part of the region, when one outbreak is discovered with 2-3 plants, I don't know now, maybe 1 year later we can detect 5, 6 or 2 more plants, but not so epidemic spread. So there is a major difference in this. Which could be the reason of this? First of all, a less suitable climate. We said before that, and if we look to the map that I showed before, you can realize that the climatic suitability is not exactly the same of the southern part of Apulia. But also there are, in the central northern part of the region, different land management, differences in agronomic and phytosanitary practices. So there is much more control of weeds, for instance. And in addition, now there is the application of the containment measure released by the phytosanitary authorities that are much better organized than 10 years ago. And of course, this is another factor for sure contribute to this in slowing this. I'm referring to the monitoring and removal of infected plants, vector control, that in some cases is mandatory, which have the objective to slowing down the spread. And what we're observing is just that the attendant result in a certain sense. And in addition, there are data saying that in the central part of the region, the population of vector is less abundant than in the southern part of the region, and this is also a consequence, probably, among the others, of different land management and application of containment measures. Few words about the funding of the other subspecies, the multiplex. In our experience, in the Apulian experience, with the strain that we have, the ST26, that so far is unique in Europe, we are not able to observe significant symptoms, significant diseases. So the Plant Protection Service is founding the bacterium only as consequence of the monitoring program. There is even the same agronomist, technician doing the sampling. They are not able to record any presence of specific symptoms. Of course, there are symptoms of draw, of the summer stresses, but are the same that are physiologic in our condition for almond, not irrigated almonds. So there is no, if we consider that the boundary of the demarcated area for multiplex is still in progress, is because we still have a positive near the border of the demarcated area, we believe that probably is even more widespread, and probably it is a quite old introduction that since it's not causing impacting diseases, escaped always from the preliminary for the detection to be done before. Now, now we are already 1.5 years, at least after 1.5 years of eradication, and the impact in our condition appears to be negligible, but we understand that it's the same in other parts of the continent. I don't know, I read one recent paper from Portugal. I'm not able to find a report of specific major diseases there, just for instance. The outbreak was discovered also only as a consequence of the surveillance program, but so far in Apulia, no one has reported the associated disease. Beside the almonds, the risk of infection of other specified plants seems to be very, very low, occasional, because in practice, in our condition, almond is almost the exclusive host of this strain. The boundary of the affected area are not yet identified, and severe impact of in this frame, the eradication program for sure has an impact that is higher than the impact of the bacterium itself. A few words also for the strain, for the fastidiosa. Fastidiosa, this has been found just near Bari, in an area that is apparently limited but not so small, because are at least, according to the monitoring program campaign of last year, there are at least 8 square kilometers interested by the presence of this strain. We are interested. Maybe my colleague that will follow me will give some update for the campaign of this year. It will be very interesting. In some case, we observe some significant impact in the vineyards for some particular cultivars, and in cases in vineyards that are surrounded by abandonment or something like that. So situations that are very favorable to to give a high pressure of inoculum. That is the situation. You can see the area interested is on the yellow oval, and you can see the color of the background that is more dark, different from the more white, almost white background that we have on the right. The white that you can see on the right are not buildings, are not city, are not municipality, but are vineyards for table grape that with the cover of a plastic cover. It means that there is a very major district for the production of grape tables that is very important by the economic point of view, that is touched, but so far seems that Xylella didn't enter in that comprehension. That has a very high economic value, also for the income of the regional economy. That's must be taken in consideration. Sorry, what happened? Okay. Oof, sorry. Can you go back a few slides, please? Go back. Okay. Thank you. So this give you a better idea of the situation. On the left, there are a few scattered vineyards, but surrounded by situation like this. That is very favorable to the spread, with some isolate almonds that quite often are infected. They are the real reservoir of the bacterium responsible for the infection of the surrounding vineyards, while in the compensatory of high value for table grapes, the situation is quite different, and we strongly believe that this condition is a condition that is hostile to the vectors. So in case, we hope not, but in case the bacterium will get into that comprensorium, we expect that the spread there will not be epidemic. So by the practical point of view, we would feel much more comfortable if the regulation will allow, in this condition, just a strategy of containment instead of eradication. Because the eradication, if the bacterium starts to spread in that area, will be devastating by the economic point of view, and we will meet with a strong opposition of the farmers. Strong opposition of the farmers, even worse than what occurred with olives years ago in Salento. But also looking to the general situation of the fastidiosa in Europe, we can see that as in Majorca, after 20 years at least of the presence of this bacterium in the vineyards of Majorca, they classified the incidence and impact of Pierce disease to moderate or low level. But also in Israel, for instance, I exchange messages with our colleagues in Israel that is in charge for the search on fastidiosa vineyards, which is the impact. We don't know yet, but there are already 6 years that they discovered this. We don't know yet means that at least there is not a devastating epidemic spread of the disease, at least of the Pierce disease. So this is the situation that is quite complex of our outbreak in Bari. And close there is a X missing. There is a multiplex spread that is almost touching this. Just a kind of provocation. Not all bad news are necessarily bad, maybe, because regardless the regulation, evidence suggests that competition between strains may modulate disease prevalence by influencing strain dominance in both host and vector. For instance, in the southern part of the island of Majorca, where the presence of multiplex is higher, the incidence of Pierce disease is lower. This because probably this impact on the infection of the vector population. So maybe there is a kind of indirect cross protection made through the vectors by the presence of multiplex. This is my last situation. So the complexity of the situation since the 2013, the picture that we have changed a lot. So the complexity of the situation suggests the adoption of targeted and case-by-case adapted management measures rather than a one-size-fits-all approach. Is it time to consider revising the regulations? So that's so from what I understand this morning from Giuseppe, from Kering, I think that work is in progress. So we may think that in 1 year or a little bit more, maybe something will be changed. Thank you.
Thank you, Donato. Yeah, maybe you can stay here with me. And so as you've already introduced, Francesco Palmisano then from the Phytosanitary Observatory of the Regione Puglia.
Good morning, everybody. I illustrate to you the situation Xylella fastidiosa in Apulia region. This first slide show the first individuation of Xylella fastidiosa in Apulia, in particular in Gallipoli. After the situation is this, in which we have several monitoring campaigns. In 2013 we have outbreak in Gallipoli, in particular any plant positive in province of the Lech. After in 2017 other plant positive here in province of the Brindisi. After up in north of Apulia and involved also the province of the Bari. And in 2023 the situation is this with other single plant positive in monopoly near to Bari. So in this table you can see the number of the activities monitoring performed to the phytosanitary service, Apulia phytosanitary service. Each row show you the number for a particular, in any case, for example, 2021-2022 number of olives sampled are 188,000, a number very high, very intensive activity performed to phytosanitary service. So in the 12 years of the activity, the number plant sampled of the phytosanitary service are about 1,040,000 plants. Very no comparable in other part of the region and the Europe. This intensive activity performed. So this is in this number regarding in part the first outbreak inaugurated in Apulia region, the outbreak ST53 Pauca, illustrated in this slide. But in 2025 the situation changed in Apulia region for Pauca, because in these years, over the number 53 positive in these years, we have sampled about 100,000 samples, and in this activity we have individuated other outbreaks of Xylella fastidiosa Pauca in Apulia. Each point indicate a new outbreak, and each outbreak is indicated the number of aerofit number with the number in which communicate the presence of the new outbreak in Apulia. One outbreak in Modugno, Giovinazzo, Bisceglie, Minervino, Murge, and after the last outbreak is Cagnano Varano, very in north of a part of the Puglia. So this outbreak, this is outbreak of the Pauca ST53 in Salento, and this the outbreak in Bari, indicated in 2024. So okay, this is an example of the activity performed of the phytosanitary service in Apulia. It's not Minervino Muge, erroneous, but Cagnano Varano. The first activity is situated the infected plant after to notify the presence the new outbreak of Eurofit in EU member other phytosanitary service of certain member states. So after we have established the new demarcated area in Cagnano Varano, in which we have infected zone. Infected zone are zone over 50 meters around of a plant infected, and the buffer zone, the area around about 2 kilometers and 5 around the infected zone. After the eradication measure, in which to eradicate uprooted the plant, the specific plant located, seated in infected zone. Very intensive activity. So after we delimited area to eradication measure, reinforced surveillance in 400 meters around the plant infected zone is performed. And after, when completed this activity, change the area investigated, involving also the whole buffer zone. So in the number very high, in few weeks, the phytosanitary service collected around 2,000 samples, and in which we have individuated 153 positive plants. Other outbreak individuated in Apulia, Xylella fastidiosa. In Apulia, the phytosanitary service conducted the monitoring not also to the plants, but also vectors, not only in the demarcated area, but also in the demarcated area. And in this case, in free area, we have individuated a vector positive in Agro di Trigiano. In this after, the area is involved in one monitoring activity on plants, and individuated 4 plant positive of the almonds. After we, the CNR conducted the characterization of the genotype and individuated in this area the presence of Xylella fastidiosa ST1. Don't change. Okay. The first activity of the demarcation of the area, we have the infected zone and buffer zone, and together is the demarcation area. This is also over the picture of the infected plant of the almonds in this area. This picture of the grapevine infected plant in the area, and the short distance between the demarcated area of Pauca and Fastidiosa suggests to identify the species presence in each new positive plant. Each new positive plant is characterized by protocol UPA to individuate the type of the subspecies. So after intensive monitoring activity around Triggiano is performed to individuate other positive plants, and the actual situation is this, in which we have in this area the co-presence of 3 subspecies. We have fastidiosa, multiplex, and pauca in this area. The plant collected in 2024 are 44,100. The positive 406. Fastidiosa 30 positive fastidiosa 339. 58 multiplex and 9 of the pauca. This is are the results of the monitoring activity in 2025. We have a prevalence of the multiplex in this area. So other subspecies individuated in Apulia is the multiplex. This individuated is obtained by monitoring activity on the plants in free area. Also in this case, characterization the multiplex ST26, demarcated area, and also in this case, eradication measure to eradication of the bacteria. This is situation now, very large area in which presence the Xylella fastidiosa multiplex and other and for the phytosanitary service use the compensation, permitted compensation of the hours, farmer that involved in eradication and containment measure. For example, in the case of the vineyard of a table, each hectare uprooted about 58,000 € for each hectare. And also not also compensation for damage, but also compensation for cost of extirpation. And in this case, the cost is 3,000. For this table, summarize the entity of this compensation for the hours or farmers involved in activity of the eradication or containment. So other okay. Okay.
Yeah, no, I mean, yeah, it's more like this.
Okay. Okay, this is are the entity of the eradication measure for the last year area in Triggiano. For approximately are uprooted 3 hectares of table grapes, 3.5 hectares of wine grapes, and other plant. Very intensive activity in this area to eradication the fastidiosa and other subspecies. So other activities monitoring vector insect. The target of the monitor insect are 3 species: Philenus spumarius, Philenus italosinus, and Philenus campestris. The life cycle of the insect is illustrated in this slide. In February, each egg hatching. In spring, in first part of the spring, development stages of the juvenile phase. in late spring adult emergency, and in summer the insect pass from herbaceous host to olive plants, and in autumn oviviviction, one cycle in each year. In Apulia region, phytosanitary service conducted the monitoring service to vector. In this slide are illustrated the site involving this activity. The choice of this point is the single point is done considering the spatial distribution of the infected plants, the proximity of the major transportation infrastructure, and the land characteristic of landscape altitude. All territory of Apulia region is involving this activity, including not also the marketed areas, but also the free area to Xylella fastidiosa. This is the scheme of the example of the activity, illustration of the activity performed in monitoring vector, juvenile stage, adult stage, and the insects collected are analyzed to verify the presence of the contamination of Xylella fastidiosa, and in the case the positive, the monitoring, the intensive activity of the monitoring to individuate the positive plant is conducted. In this specific website, whole information, the act of the eradication, act the demarcation, data regarding the monitoring activity, both vector and the plant, are reported, and other activity. I have illustrated you the activity to perform, to individuate a new outbreak, to eradication the bacteria, a contaminating bacteria. Other activity is extraordinary plan for the regeneration olive growing in Apulia. This activity is regulated to the interministerial decree published in 2020. In this slide, we have the main topic for this interministerial decree. So, for example, the first each activity, for example, vector control, elimination of inoculation source, for each activities, there is a specific budget. Other activities, replanting olive trees, conversion of upper clips, and other activity, including the communication information. The very part important in this program is replanting olive trees in infected area, and the measure aims to promote the recovery of potential production through financial support for removal plant trees that are not longer productive. The total measure fund allocated is 80 million euro, committed 64 million, and beneficiaries about 3,000. Other activity in this program of communication information, in this activity is included the project Regenesi. In Regenesi project, the all information obtained in the Apulia region is used to information other office, other phytosanitary service in third countries. And in these slides reported the possibility of the download of the first briefing. So, in conclusion, the measures are conducted to combat the spread of Xylella fastidiosa in the Apulia region, considering the monitoring regarding the monitoring of territory, early detection infected zone and detected infected plant, and after eradication and containment and control in the vector. Also, other measures are conducted to regeneration, destroy olive growing potential, including the interministerial decree of March 2020. So, considering the different epidemiological characteristics of Xylella fastidiosa pauca, Xylella fastidiosa multiplex e fastidiosa, it will be preferable to revise the EU legislation to provide the mandatory measure ponderated or specific, distinguish this measure, this mandatory measure, in considering the different damage or different symptomatic determined by each type of subspecies. Okay, I have a conclusion. Thank you.
Thank you, Francesco. And please, please take a seat. Yeah, you can maybe share a microphone here. We won't take too much of your time, but maybe just a couple of follow-up questions before we go on a break. So there's intensive monitoring going on in the region, so you have a lot of experience, and thank you for having shared that experience. One thing that you mentioned is that you've realized that there's no one-size-fits-all, and even within the region of Puglia, there are different speed of spread of the disease, and therefore different realities even in the region. When it comes to a potential solution, like let's say a biopesticide that may come onto the market, do you expect that a single product could nevertheless be used in all cases? Let's say in the region of Puglia, or even within the region of Puglia, you would expect different types of products necessary to contain the spread in different areas, even including within Puglia? Or would you say the product could have a much broader reach? What's your expectation?
Concerning the product, I think that the availability of efficient product, it will be could interest everything and everywhere, regardless the different speed of spread. So I think that it's more important to adapt case by case, to focus also on the practices of management in general. So not only looking to the pesticide or whatever, to the product that you can apply. So that's my idea.
Yeah. And on genetic resistance as well, because obviously that probably takes a lot of time to develop genetic resistance and to have that in reality in fields. But do you see anything coming? Is there any research that is happening in this area that can have a substantial effect? And again, how much can that adapt to different realities, including within the region of Puglia?
Okay, so we have to look. As we said, the situation is complex. There is not only pauca, there is not only olive. We have to keep the attention, because still we don't know, as I said, the real impact of the fastidiosa, but we must keep open our ears also on the evolution of this subject for the grapevine, especially. And also in some parties, it's not the case of Apulia, but this also applies to the almond, if we look to the situation in Spain. So for the in case of olives, there are several programs of research to develop germplasm resistant to Xylella in Italy, at least, because there are several projects. In particular, there are 3 consortiums funded by the Minister of Agriculture now that are focusing on a program of genetic, of breeding or development of resistant germplasm. In the case, I'm more informed in the program that is going on in my institute that is coordinated by the colleague Maria Saponari, and for that there are already promising things underway, because there are, it has been differentiated in different line of research, but from each one of these there are with a different step, but there are, I don't know, in the case of the selection of spontaneous seedlings that has been selected in the territory, now there are two selections that are for which has been initiated the ITER for the registration of these two cultivars. There is the first activity that started 10 years ago, as was mentioned a little bit by Giuseppe Stancanelli before. Quite soon we'll release the results, and we are confident that among them there are 5, 6 maybe cultivars that may have characters of tolerance. That is not a final solution, but it is an help. And then, but the program of the traditional breeding took much longer time, and we'll see in the future.
Yeah, and maybe a comment then also from Francesco on that, because obviously that takes a lot of time. So the biopesticides or all these kinds of products are important in the meantime, right? And probably may be able to get to market quicker, but they still need to interact, right, with developments in genetic resistance. So maybe a comment on that as well.
So it's very, very important to have the new germplasm to the resistance and tolerant, because it is new. Important have it is in very low time, because to substitute the old cultivar in Salento with other new cultivar, all the results of the research.
Okay, thank you so much. We have a question here in the front. We'll take that one, and then I promise I'll let you have a short break. Okay? Please briefly introduce yourself.
Stefan.
That's true, you're already on.
Yeah, I have one question. When I see the area where you have the different subspecies in the same area, is there any risk that there is some horizontal gene transfer between the different subspecies? Because the bacteria, they are with the insect, it's the same insect, taking the different subspecies to go to different hosts. So there is this risk scenario. But for the moment, we don't know. Huh? I'm afraid of that.
Yes, but if I will understand you mean the risk or a combination? Yes, the risk there is, but of course, but there is a risk, but at least so far we haven't found any case of recombination or any case, at least so far, of double infection in the same insect, in the same so far, but you never know.
Okay, Donato, Francesco, thank you so much for having shared your insights. It's time for a short break, 20 minutes, okay? One thing, if you're beginning to feel tired trying to understand the English, there is interpretation, okay? So after the break, if you like, you can also have access to that, all right? See you in 20 minutes.
champagne. Ah ouais, moi j'ai tout basé sur la viticulture avant d'arriver en Autriche. Et puis en Autriche, bah, il faut que je fasse des projets sur tout. Et comme j'ai un peu le son italien, les olives, tout ça aussi.
Okay. Well, I have to say, congratulations on getting. I didn't expect you would be already coming back into the room at 46. I did not expect that. So you are way, way, way beyond expectations. Thank you so much for that. We will get started then with the second part of the event, where we will learn a lot more now about the BIOVEXO project and the results that came out of it. So, I would like then to invite to the stage to present the project Stéphane Compens, who you've heard from Stéphane already from the AIT, Pasquale Saldarelli, research scientist at the National Research Council of Italy, and Enza Don Giovanni, who's a researcher at the CRSFA Basile Caramia. Please welcome Enza, Pasquale, and Stéphane to the stage. We're just missing Enza. It's all good. Enza will be here in a second, I'm sure. In the meantime, I propose then that we first hear from the BIOVEXO project in video. We have a short video to show you, so we can go for that one.
In southern Europe, olive and almond trees are more than crops. They are heritage. They are landscapes. They are livelihoods, passed down from generation to generation. For centuries, they have shaped the culture, the economy, and even the identity of entire regions. Communities thrived around their harvest. Generations found dignity and work in tending these groves. But in recent years, this legacy has been under threat. The spread of Xylella fastidiosa has devastated entire regions, leaving behind fields of lifeless trunks where once stood thriving orchards. The bacterium destroyed centuries-old olive groves in Puglia, reshaped the landscapes of Majorca, caused the loss of traditions, rural economies collapse, Mediterranean beauty fades, tourism declines, businesses struggle, and young generations, seeing no future, abandon the countryside. What remains is a wound on nature, culture, and communities. When the BIOVEXO project began in May 2020, our mission was clear: to develop sustainable, effective, and environmentally friendly ways to fight back. Protecting trees, farmers, and the traditions that depend on them. over the past 5 years, a team of 11 partners from 5 EU countries, coordinated by RTDS Group and the Austrian Institute of Technology, has worked side by side to make this vision a reality. From an initial selection of 6 promising candidates, BIOVEXO developed 3 innovative biopesticide solutions, 2 plant-based extracts and a beneficial bacterium. These natural agents have been tested not only in laboratories, but also under real-world conditions in the orchards of Apulia, Alicante and Mallorca, where the stakes could not be higher. By integrating these solutions into broader phytosanitary and agronomic management strategies, and through large-scale trials, we are beginning to see encouraging results, reducing the pressure of Xylella, protecting newly planted in Europe. BIOVEXO is proud to contribute to this ongoing effort. And the story does not end here. It continues in every orchard that is protected, in every farmer who can carry on their legacy, and in every community that can look to the future with hope. To follow the journey beyond the project, visit our website and stay connected through our social media channels.
Okay. Yeah, you can clap a video. Yeah. You can. Brilliant. So, Pasquale, Stefan, Enza, you have 40 minutes to present us the BIOVEXO project.
So welcome again, everybody. I'm going to explain. I will explain what we have done in BIOVEXO during this 5 years project, from the lab to the field. How we try to develop different biopesticides. But first of all, this is a project granted by BBIGU, it's now CBEGU. It's a 6,600,000 euro project with industry like Globachem, Domka, Assess Bio, some RTOs, and also academia. And our objective were to develop some biopesticide to reduce the disease pressure caused by Xylella. Disease severity, and also to protect on the long term olive cultivation, but also almond trees in Mallorca. And to secure jobs, because farmers lost a lot of jobs because of the Xylella disease, especially here in Puglia. And for that, we started not from scratch, but we had some solution in our pipeline, in our different laboratories, with some beneficial bacterial strain that can be antagonist or not to Xylella, some plant extract, microbial metabolite, and also an antipathogenic fungus. And the idea was to test this solution as preventive or curative against Xylella directly, but also against the vector. So to have different approach for sustainability. Sorry? Okay, like that. And for that, we had different work package in the project, starting to evaluate at a small field scale trials, some biopesticide targeting the vector or targeting the Xylella, also to upscale and formulate the different biopesticides, to test on the large field scale trials, and also to optimize delivery and to assess about the sustainability of the product and the toxicology and the toxicity. The result obtained so far, what we got. We upscale and formulate different biopesticides. We start with different one at the beginning, but we select some to go further into testing on the large-scale field trials. We tested spray or drip irrigation on also endotherapy, on almonds or on olives against Xylella. We analyze also the life cycle analysis, and we determine part of the mechanism of action. When we know that there is some biopesticide working or not against Xylella or against the vector, what are the mechanisms involved? And yeah, we start with small-scale field trials, different solution in olive orchard, in Puglia, on almonds and olives in Mallorca. We identify the best application method, how to apply the different product. Some products should be used with drip irrigation, some of the foliar spray, some of the endotherapy, and as curative or preventive. And every 6 months, we monitor by qPCR the Xylella population, but also the symptom. What we got at the beginning, we start to see something on plant vigor. There was no solution as curative, and as preventive, there was no symptom at the beginning. But we select some solution named formulated PAGN, onion extract, and a bacterial strain, Pseudomonas PV8, for further testing. For the insect vector, we made some lab tests to confirm efficacy on the laboratory condition of some substances, identification of the best application method, so interval of application, the rate of application, the dose range, before to select some V biopesticide targeting the vector for further application. We started with 3 biopesticides targeting the vector. But at the end, we select only one, which is named Sankari from Drobachem. And you will see some results later on about this biopesticide targeting the Philaneus spumarius. And here, on the laboratory condition, you have the control, so untreated insect. You have several dilutions of the Sankari product, and you can go up to 100% mortality of juvenile or adult insects after application of the product Sankari, on the laboratory condition, first. And the mortality was assessed different minutes after treatment, 1 minute, 2 minutes, 30 minutes, 60 minutes. And what we have done just after, with the different X biopesticide, remember, targeting Xylella symptom of the population, and the V biopesticide targeting the vector, we needed to upscale the production to apply sufficient amount of biopesticide in large-scale field trials. And also to optimize the formulation, to put some adjuvant, also to increase the shelf life. And it was a lot of work by industry. It doesn't work. And what we have done so far on optimization of formulation. For the X biopesticide, we got 2 products based on onion extract from Domka, Trophic Liquid Evolution IT, that can be applied as foliar or drip irrigation, or trophic powder endo for endotherapy. The strain Paraburkholderia phytopheromone strain PAGN that has been shown some nice result in California against peel disease by Stephen Lindo was upscaled to 6,000 liters, formulated for extended shelf life, suitable for large-scale field development. And for the V biopesticide, robust formulation of Sankari with packaging and validated storage stability. And then what we have done, so this took several years already, since the beginning of the project. And I told you this project is 5 years. It has started in 2020, will end in December this year. Then we evaluate this product in large-scale field trials. And now Pasquale will show you this result, what we get on the field condition. We have a lot of different plants. Then we will talk about IPM strategy, and I will come back after for mechanism of action and toxicity. Pasquale.
Okay, thank you. The worst that could happen to me is to work in the field, from the lab to the field, but it was a challenge to to me to work on this. And I coordinated these large-scale field trials that, as far as I know, this BIOVEXO represent the largest project working on such a big number of plants. We work on more than 100 of plants in the fields. And we tested these two biopesticides for Xylella, against Xylella, and against the vector in olive, in almonds, in Italy and Spain. This is a real-life evaluation, because I learned that finally you should move in the field to evaluate the efficacy of every product, and in two different approach, a curative and the IPM approach. This is a summary of the field trials that we have ongoing, in Italy. There are two curative field trials in Oria and in San Vito dei Normanni, so in the epidemic area. These are plants already infected at the start of the trials, 30 years old olives, while these were planted by us during the BIOVEXO project in San Vito dei Normanni. In Francavilla Fontana and in Squinzano, we have another two field trials which deals with an IPM approach, an approach that combines the use of the biopesticides against Xylella and the biopesticides against the vector. These are the field trials in Mallorca. These are made in olives, but we give up with these trials because they were not very infected. We moved and have the better results in field trials on almonds, and you can see here the field trials in Santa Maria del Camí. Sorry. The field trials of Oria. The cultivar is susceptible, Cima di Melfi. These are the treatment that we tested in the fields. Plants were already infected with symptoms at the start of the trial, and the two biopesticides were the onion extracts and this formulated PSJN, these endophytes, with 8 treatments per year. The results that I'm going to present are related to a period of 4 years, because you need this period to get at least significant results in this kind of plants. Plants are 120 plants and randomized in a classical randomization that are applied in field trials. This is the field trials in San Vito dei Normanni, 168 plants against randomized, the same treatments, the same products. These plants have no symptoms at the start of the trial because we planted this in 2021, correct, Enza? 2021, so we follow it. These are, to me, these are very precious, these orchards. I would like to continue to maintain these orchards beyond BIOVEXO, because we know the history of all these plants for 4 years, because every 6 months we monitor Xylella by symptoms and also by quantitative PCR. So we know the evolution of the epidemic in the field for all these years. And another points that I want to highlight is that these curative trials have been carried out without any control of the vector. So the plants are exposed to natural infection without any control of the vector. These are the results in Oria. We observed an increase of the symptoms throughout the years. These are of distribution of symptoms. The owner pruned the plants in 2 different times throughout the trials, and we consider this was an heavily pruning that was done last year, and we consider this as a part of a possible IPM approach. We evaluate two kinds of symptoms. New symptoms, that means the symptoms occurring in the ear, and desiccated shoots or old symptoms, so symptoms that belong to the previous year, so that means branches without, that have lost the leaves throughout the years. And we also evaluate another parameter that I'm not going to present, which is plant vigor, which is very important for one of our two products. Okay. These are the results in Oria, and you can observe this is a sort of way of expression of symptoms, the area under disease progress curve. It is a cumulative observation, evaluation of the symptoms in the whole period. What we observed is that new symptoms in Oria were significantly reduction as by this statistical analysis in this case, while desiccated shoots, they were not significantly reduced. The other field trials is San Vito dei Normanni. Of course, we observed an increase of symptoms, particularly the last season was very drought, which was very hard for this trial to maintain. It's a curative trial. And here we again observed a reduction, significant reduction of new symptoms and old symptoms for one which were significant for one of the two biopesticides we tested, which is the formulated PSGN. I remember that, as Stefan said before, formulated PSGN is a Parapurcoldiria phytophyrmans strain PSGN that was already described in California, working in Pierce disease. It was described by Stephen Lindov in California. He's already working on it. Oh, cazzo. No. Perché? Okay. The bad news is that we observed no statistically significant difference in Xylella concentration across the years. So there is a sort of reduction by the formulated PSJN, but it's not significant. So we were not able to eliminate the pathogen from the infected plants. That is a limit, but I think it's a limit of the particular pathosystem that we are dealing with. So we do not guarantee. This is particularly important because people, as Donato commented before, try to propose some mitigation of the disease in the buffer area. That is not possible to use pesticides that do not eliminate the pathogen from the infected plants. I was not so very. I wanted to expand our information about the observation about these plants, not only limited to the visual observation, because what you have seen before was related to symptoms observation on a scale to 0 to 5, and we wanted to, and we are also introducing this evaluation in all our activities in the field. We want to introduce some numbers, some real numbers in the evaluation of the plants infected by Xylella. One of these is the use of the evaluation of the stomatal conductance. Xylella induce a draw stress in the infected plants. That means that block the xylem and also the plants react by closing its stomata. We have also published this data on a previous paper, and the instruments to evaluate this is the porometer, is the instruments that evaluate the opening of the stomata in the plants. And then we can compare the status of the infected plants against the healthy plants. If this difference, of course, is close to zero, that means that the infected plants is not suffering because their stomata are similarly open as the infected plants. And what we observed in this case, it was a confirmation of the efficacy, I would say, of formulated PSJN. This difference was significantly different in plants treated with formulated PSJN on a number of plants. These data are related to a number of plants of this field trial. If stomata close, the temperature of the plants increase because there is no exchange, gas exchange. So we also evaluated, in contemporary, we also evaluated the temperature of the canopy of these plants of San Vito dei Normanni in different situations. We observed against that plants treated with the formulated PSJN are colder than the untreated plants. So confirming the data from the porometer, from the stomatal conductance. This is a final summary of the progress of the symptoms throughout the time from March 2023 to July 2025. Every line on the graph corresponds to the full line is correspond to the formulated PSJN, while the break lines corresponding to the plants treated with onion extracts. We observed in Oria that new and old symptoms were reduced in treated olives in comparison with untreated olives. So these data are the treated plants subtracted by the untreated plants. In Oria, this reduction was of the 11.3% for the formulated PSJN and 10% for the onion extracts. And this is the situation in San Vito dei Normanni. In these young plants, we observed a reduction of new and old symptoms in olives treated with the formulated PSJN for the 27% and 25% respectively in new symptoms and old symptoms, as I described before. These are the results of our field trials in Mallorca on almond trees. This is the orchard in Mallorca. And on July of this year, our colleagues in Mallorca, they evaluated the symptoms on the same scale that we use in Apulia. And they observed again that formulated PSJN reduced the symptoms in plants treated with these products and also with the onion extracts. Even also in Mallorca, as in Apulia, the increase of positive samples increased during the time as evaluated by quantitative PCR also in Mallorca. So the highlights is that our formulated PSJN slowed down symptoms on young plants when application was started at the early stage of infection, particularly in olive. These products improves the stomatal conductance and plant vigor. We have also some trials ongoing to evaluate the vigor induced by this plant growth, because PSJN is a plant growth promoter bacteria. Reduces symptoms in almond, and similarly in onion extracts there are reduction of new symptoms in olives and reducing symptoms in almonds. We confirm that none of the two biopesticides lower the Xylella population with statistical significance in infected plants. And I think I stopped with this, so Enza.
Good morning to everyone. I linked the presentation of Pasquale. As said Pasquale, in two curative trials in Oria and in San Vito dei Normanni, we have applied specific measures for the control of the bacterium with applied PSGN in spray and onion extract in drip. But for the entire period of the trial, from 2022 to 2025, no specific measures were applied for the control of the vector. While in this period, we have evaluated in both trials the density population of Philaenus spumarius and also the percentage of the insect harboring the bacterium. As evident in this slide, in both trials, in San Vito dei Normanni and in Oria, the density population is very high, but in particular, the percentage of positive insect to the bacterium is in 2022 and in 2023 more higher than the mean value that we have obtained in other sites that we have monitored in Lecce and in Brindisi province. So this high density population and this high percentage of insect positivity to the bacterium probably influencing the transmission events. And so we have not the possibility to verify the reality effect of the tested product against the bacterium. From 2022, we have set up two field trials in infected area, one in Squinzano and others in Francavilla. Very big field trial, about 1 hectare, with about 400 olive trees that were planted in 2022, and the plants were exposed to natural infection of the vectors. In this field trial, we have evaluated the effect of the both selected products, PSGN and onion extract, against the bacterium, and they are integrated with specific control measure against the juvenile stage of the vector using the product based by pelargonic acid, Sankari, and also the specific application against the adult of the vector, also using at different rates a biopesticide based on pelargonic acid, and compared with plot treated with chemical insecticide, acetamiprid, this authorized product for the control of Philaenus spumarius. In this plot, each treatment against the bacterium were repeated a lot of times, 18 times, and each plot consists of 16 plants. As in curative trial, we have carried out 8 treatments each year, starting in March up to November, and the application Okay, okay. The application against the juvenile stage of the vector were applied during the spring when the juvenile stage reached the third, fourth instars. Two applications in two big plots, about 2,015 square meters, reported in the slide in blue, and this big plot were compared with a big plot untreated for the juvenile stage. In each big plot, 14 transect is a method to evaluate the density population of the juveniles were considered, and we have evaluated the number of the split, the number of individual in each split. Okay. Only in one trial, in Francavilla, we have evaluated the effect of GROB-BP applied against the juvenile stage, and we have obtained good efficacy with an Abbott index ranged about 50 to 54%. Lo fa lui. Okay. Regarding the adults, we have treated four plots, two plots with organic product, pelargonic acid, and two plots treated with chemical insecticides. The application were carried out when only one individual of Philaenus spumarius were captured among the 32 sticky traps placed along the edge of the untreated plots. And the application starting when the adults move from the ground vegetation to the olive canopy. And then we have carried out application with the GOBP each 7 to 12 days, while with acetamiprid each 12 to 15 days. Okay. No infected plant affected 3 years after the starting of the trial. We have collected a sample from the shoots, and we have evaluated the percentage of infected plants within each plot. As evident in this slide, no infected plants were detected in plot treated with chemical insecticides 3 years after the start of the trial. No infected plants were recorded in formulated PSGN untreated against the adult of Philaenus spumarius, with an Abbott index of 100%. An evident reduction of the percentage of infected plants were obtained in plot treated with formulated PSGN and onion extract with an Abbott index ranged to 50 and 70% in plot treated and integrated with product based with pelargonic acid for the control of the adults vectors. Regarding the field in Squinzano, we have carried out the application against the juvenile stage and the adults, adopting the same method adopted in Francavilla. In this case, we have not observed difference among the treatment against the bacterium, formulated PSGN onion extract, as compared to untreated check in plot untreated against the vector, while good results were obtained when we have integrated the application against the bacterium and integrated against the vector in both conditions using chemical insecticide with an Abbott index of 18%, also integrated with chemical organic product with an Abbott index of about 50%. No result were obtained in this case using onion extract, regardless the treatment against the adult vectors. In synthesis, using saccharine, pelargonic acid, for the control of the juvenile stage of Philenus spumarius, we have obtained an effectiveness of about 50%. And integrated pest management can reduce the percentage of infected plant, in particular when we have used a formulated PSGN, and we have integrated this treatment against the bacterium with a specific application against the vector. In both the conditions, both using chemical insecticide and also using organic product. Inconsistent results were observed using onion extract. No effect in one trial, while a strong effect in other trials in Francavilla, where the percentage of infected plant was reduced about 76% when it is integrated with using of saccharine based with pelargonic acid against the vector. So in synthesis, these results appear promising. The integration of in preventive trial of specific measures targeting the bacterium, but also the vector, can reduce the progression of the infected plant and the disease. But I think a further long-term study is necessary to verify the effectiveness of this integrated combination in the long periods. Thank you to it all for the attention.
This is not finished, sorry.
5 minutes?
Yeah,
Stefan?
sure. So we show you some results from the lab to the field, and now I would like to come back to the lab to determine what was the mechanism of action with the different substances, the different products. So what we assessed first, if there was antagonistic activity against Xylella on petri dishes, formulated PSGN or PSGN alone, there was no direct antagonism against Xylella, only onion extract. But we also evaluate the effect of the biopesticide, the different biopesticide, on the plant immunity, the plant transcriptome, the gene expression of the plants after application, also if our biopesticides change the plant microbiome, the different component of the plant holobiont of olives, if there was an increase or decrease of other kind of microorganism associated with plants, and also on the insect microbiome. And we evaluate also if our solution were persistent or not in the environment, like with formulated PAGN. This is just one example of result from the lab with plants under greenhouse condition, and what we saw with formulated PAGN. When we applied formulated PAGN on plants, there is an increase of gene expression related to plant metabolism, carbohydrate metabolism, photosynthesis. And when we put the Xylella on the plants, it looks like the plants have less stress when the plant is pre-treated with formulated PAGN in comparison when you have plants treated only with Xylella, where you have a lot of stress in the plants. And we have done also some other things on the plant microbiome. We don't have enough time because I have now only 4 minutes, I think. And what we wanted also to assess, we had some nice results from lab to the field. Reduction of symptom, no reduction of Xylella population with the X biopesticide directly as curative. Combination of different products with V biopesticide and X biopesticide on IPM strategy when you saw a reduction of the infected plants. But what is very important also is to assess the sustainability of the product and the toxicology and the toxicity. So we analyze the life cycle of each product, if there was less energy cost when we make the product compared to existing product. Sorry. And so we have done also toxicity evaluation on the human, on the environment, selection of the best solution. And we know that our products are non-toxic. And in a nutshell, to finish, yeah, we formulated PAGN. We have a reduction of some symptom in San Vito de Normandie. After 4 years of application, we know it's improved also plant vigor. We saw better green plants. Onion extract also improves tomatal conductance, plant vigor, reduce appearance of new symptoms on olives and almonds. Some carrier reduction of the juveniles and insect adult. And in IPM strategy, we saw also a reduction of percentages of infected plants when we combine with chemical solution or biological solution. There is, unfortunately, no product yet on the market for the farmer. We are close to market entry, but have in mind that this took 5 years to start from the development, innovation, and to get this result from the lab to the field. And I would like to thank you very much for your attention. There is only my name written, but we are all here from BIOVEXO together with this result. Thank you very much.
Thank you so much. So that was some insight into the BIOVEXO project. We have, I think, time for a couple of questions before we break for lunch. So please briefly introduce yourself.
Yes, thank you very much. I'm Fabio Gatti from Wageningen University, social scientist. So I was very curious about the product that you developed, and I had a few questions in mind. One, I was just curious, did you try also to apply it on old trees, like centuries old trees, rather than just like young, and see if...
No.
And see if there is an effect. And then, I have a second question, but maybe it's a little bit provocative. Why do you call it a biopesticide if you don't see a pesticidal effect? If you don't see a decrease in the density of the Xylella in the tree? And you, yeah, to me, this is just like, how does this compare with other products that have been used in the region that show a similar reduction of symptoms, and in some cases also a partial bactericidal effect? So does this product outperforms those? Thanks.
It depends of what you intend as a bactericide effect, because one of our products, onion extracts, has a bactericide effect in vitro. So you should be more precise if you ask. If this has a bactericide effect, but in the plant, I want to see if it reduces the population size of the Xylella. So this is the first. No, we have not tested monumental trees, but our trees are 30 years old. The one in Oria, they are 30 years old. We have not tested initially, perhaps also, I think, as my comments, consideration, that more can be done in the way of application. So a monumental trees is a difficult trees. There is another part that we did not mention, that we tested endotherapy with this product during the project, because endotherapy could be applied perhaps better on monumental trees, but unfortunately it was not better, the efficacy of the products by endotherapy with respect to the spray application or drip application as we did.
If I can add something for the question regarding the biopesticide, we have seen both effects, biostimulant effect on plant vigor, and for biopesticide, even if you don't reduce the Xylella population, but you reduce the disease symptom, it's still a biopesticide in the definition. But we have seen both effects, plant vigor, reduction of symptom, according to the product, of course.
Thank you very much. Thank you for the two speakers. Very, very good presentation. Very interesting results. My question is, what is the possibility for BIOVEXO to share the results or part of the results with EFSA for the risk assessment? I understand that you are bound to the industry, you need to publish, but maybe something is already published, but...
That is a sensitive question. That is a sensitive question. Indeed, the difficulties in working on this such project is to work with companies. As a research scientist, I suffer in this condition. I should be honest with the people from the companies that is in the hall. Because there are conflicts, there are interests also, honest interest of the companies in this stuff, in this story. We are trying to publish these results. However, we are trying to publish these field trials results. The paper is already written. Now they are partner are reviewing this for a final. Also because as a scientist, I feel the obligation to share these results, even if they are not completely a miraculous solution, but perhaps someone after me could use this data to better modulate the use of these products. Regarding the data, if we publish these field trials, of course we should publish even in the Zenodo, in the EU repository, we should publish the Excel 5 that reports all our field observation, quantitative PCR data. So this should be published, and then perhaps this could be useful for EFSA to have numbers to work with. Did I answer to the question?
I think Stefan, you wanted to add something?
Also, I forgot to say that the results, the publication with the curative approach is on my computer. I'm a bit late, a few days. It will be submitted soon.
Okay. Final question? No? We're good? We're hungry? Yeah. All right. So thank you so much, Enza, Pasquale, Stefan, for having shared these insights on the BIOVEXO project. So we will now break for lunch. Before we break for lunch, I just have one question for you. Is everyone connected to the internet? here, like either Wi-Fi or mobile data or whatever? Yeah? Yeah? Okay, because that'll be important for after the break. But okay, perfect. So enjoy the break. I guess it's in the same place as where we had coffee, right? So, and we'll have an hour. Okay? Thank you. See you back in an hour. Okay, okay. Okay. Yeah, we can start. Huh? We can start. Yeah, we good? See? Okay. Let's get started again. Thank you so much for your diligence. Apologies for having pushed you through. But to be honest, we're still on track, and you will see, you know, when we fine-tune a few things, we'll still end at the same time. We'll make this work, okay? We'll do this together. Also, a quick word to speakers. Please do your best, all right? Please do your best to make it as short and sharp as you can so that we stick to the time. And also in the afternoon, we'll have a little bit of interaction also with the audience. So you'll see this will work. And also let me know if you feel that the lights are a little bit dim. We'll see. We need to dim them a little bit so that the translators can have a better communication and can understand better. But if it's too dim, anyway, we can adjust the lights a bit later. All right. So we will now hear from a few other projects in the field tackling the problems with pests and Xylella. So we have Maria Saponari from the IPSP CNR. She's senior researcher. We have Giuseppe Lima, who's full professor at the University of Molise, and Stéphane Compon will be also presenting the BIOVEXO project, or representing rather the BIOVEXO project, not presenting, not re-presenting either. No. All right, so first we'll be hearing then from two projects from Maria Saponari, the Bexil project and the Omi breed project. So please welcome Maria.
No? Okay, now, okay. First of all, I bring the apologies from Blanca Landa. She's the coordinator of the European project Bexil. She could not travel and attend the meeting, but she sent a few slides just to present here what has been done in the Bexil project, since this European project is also almost at the end. Next year it will be the final report of the project. I asked the technician to launch. She sent a video that summarizes a little bit the project, and then I will show just few slides regarding the complementary actions of this project with BIOVEXO. you can put the presentation, I can add only few more words regarding the topic of this project. This was the first project funded under the Horizon Europe program. Okay, sorry. Okay, sorry. The consortium is quite large. We are 28 partners from lots of countries, as you see here, not only European countries, but we have also third countries. Their participation, although is not funded by the European Commission, was considered highly important to exchange information and knowledge from these countries where the bacterium is already well established or as is infecting crops from which we need information in Europe. Regarding I go fast, as we said, the project includes different objectives, and so it's not only the curative method, differently from BIOVEXO that was targeted on curative methods and preventive methods. The project also includes the strengthening of measure to protect the European Union from the introduction of Xylella and for the further spread. So there is relevant activities on improving surveillance and modeling for prediction of the spread, and so to strengthen all the measures for preventing severe impact of the pathogen. Sorry. Okay, and the final objective of the project is to have an IPM strategies, because it's quite clear that we cannot tackle Xylella disease with only one action or one solution. So the objective of the project, as it was also in BIOVEXO, is to put together an integrated strategies to counteract the impact of the bacterium, which include the resistance of the host plants, but also action and solution against the bacterium and the vectors. One peculiar point of the project is that during the project it has been set up a network of sentinel plots. So in all these countries, through the project, there will planted a set of defined species which have the role of sentinel plants. So these include outbreak area or Xylella-free area. In the outbreak areas, the objective of this plot is to see which is the species that on which we can early detect the infection, and so a plant species that should be regarded as a sentinel plants. Whereas in the Xylella-free area, the scope and the aim of this plot is to eventually intercept introduction and establishment of the bacterium, which has not been yet detected. I will not go in depth in the IPM strategies, because Professor Gianni Giglioli in the next session will speak about it. But I will mention some aspects that are complementary to those presented in BIOVEXO. In fact, when the project was prepared, it was paid attention to be complementary and to do not overlap or redo work that was already done in other European actions. So, for example, for the vectors, the project concentrated on the use of some endophytic fungi, and this is some of the illustration. Sorry for that. I will not go in depth in the data because, of course, there is no time. But and these were both tested in the field with the involvement of the group of ENSA Don Giovanni, both for young, for the juvenile, and for the adults. Another aspect that is complementary to what we already heard this morning is the study of the virome associated with the insect. So to study if some of the insect that has manifested some alterations could be related to the presence of some viruses which could be used for our purposes to reduce the fitness of the insect. And this work is carried out, is under the leadership of Professor Domenico Bosco in Torino. As well as he is also leading the work on searching for parasitoids, which unfortunately seems not to be a good route for the control of these insects, as a low number of parasitoids, the eggs or adults, were collected during the surveys carried out during the project. And the third action that is included for the vector control is the agronomic management, and these activities are ongoing in Spain and in Italy, in grapevine in Spain and in olive in Italy, as also carried out by the group of ENSA Don Giovanni. And regarding the control of the pathogen, the innovative tools that I want to underline in BEXIL is the development of a synthetic community of microorganisms. Knowing that the Xylella has a complex interaction with the microbial community in the plants and the difficulties to control this pathogen in the plants, we know that it would be a dream to identify only one antagonist. So the idea behind this task is to find a consortia of microorganisms that can protect, mitigate the impact of the disease in the plants. So I think that next month we will start the field trials with some of the microorganisms that have been isolated either from the plants and from the vectors to be used, to be tested in the field here. And the same there is also in our project the testing of antimicrobial plant extract, whereas one of the innovative approach is the use of peptides. These were already tested in previous project, limitedly to in vitro test, but in BEXIL we are now in the field stage. And yes, I will not go, as I said, in the integration of these methods and how they are applied in the field, because Professor Giglioli will explain later. So thank you, and these are the reference of the project. I will continue now with the project that actually I coordinate. Is a national research project, and it deals with the resistance on Xylella fastidiosa in olives. It's the project Omibrid. This project has been funded in 2023 along with another two projects which target the study of the resistance against Xylella fastidiosa. We know that what is the resistance in two words. We know that once a plant gets infected by the vectors, there are some plants which react to the infection, and the bacterium, even if start multiplication in the plants, the colonization is quite limited and restricted in the canopies, whereas the susceptible plants are intended those that succumb to the infection. So as you can see in that drone, the bacterium is able to digest the pit membranes of the xylem vessel, and so to easily colonize the plants and fully invade the plants. At the moment, the mechanisms that occurs in the resistant plant, as well as which are the host determinant of this resistance, are not yet known. There is more knowledge in grapevines, where the studies have a long history in US, but very little is known, for example, in olives or yet in almond. So the starting point for this research was the evidence since the beginning of the epidemic in Salento that there were some plants that survived the infection. They are not immune. We are not speaking about immunity, but they suffered much less than the susceptible one, and they continue to produce. So in these past 10 years, lots of research have been carried out, investigated different aspects of the plants to understand if it's an anatomical difference, physiological or genetic, have a genetic basis, or the metabolic profile, or if these plants harbor a different microbial community. So far, unfortunately, the evidence that we collect is that the genetic traits associated with the resistance in olive are very limited in the germplasm. So there is only a small cluster of genotypes which harbor these traits. And exploring the natural genetic resource in the epidemic area, I mean the wild olives in the infected area, we also confirmed that if there were some plants showing resistance, not suffering from the infection, most likely they all derived, if not all, most of them derived from lecino. So they were seeds of lecino cultivars. What's the project? Which are the main objective of the project? The first task that we have just completed during these second years is the collection of a wide range of olive genotypes to collect as much as possible the genetic variability that exists in olives. And the main attention was paid to the ancient olive trees so that we can capture as much as possible the variability that occur in the Mediterranean basin, where the origin of this plant species is. Sorry. And what we will do with all the data, the genetic data that we accumulated with all these genotypes, the group, the genetician, are characterizing these plants in order to identify unique genotypes. In olives, it's quite often to find synonymous or plants that are identical but named with different names. And so the final objective is to have a map of the unique genotypes and then combine this data with the phenotyping data. For phenotyping data, I mean how these plants react to Xylella or other stresses. Of course, we cannot test 1,000 different variety for Xylella because the efforts that are needed, either if we do the screening in the field or even in the greenhouse, is huge efforts and take long time. But on a small subset of cultivars, we are producing this data so that they can be matched with the genetic data to find which are the conserved sequences across the different cluster of cultivars. We are not working only on Xylella stress, but this abiotic stress, but also on abiotic stress like determine which is the cold resistance of the cultivar. This is a quite easy phenotypic data to acquire because it's quite fast, as well as we are doing also a work to determine the response to drought stress, because it is another important factor nowadays. The other points important from the project to identify which are the genetic determinants associated to the resistance is to use segregating population. So we have population crosses derived from the resistant lecino and the susceptible Oliarola, and we hope that in the near future, we can get some information by characterizing the segregating characters in the population. All these plants are genetically characterized, so once we know how phenotypically these plants react, we can again associate the genetics to the phenotypes, and so in the hope to associate and to identify the genetic region of the plants that are responsible for the resistance. It's also important nowadays to have an interaction between the different approaches that the new era of genomics offers. So one of the important task of the project is to combine different approaches on the same set of plants, and this is very important. Then all these tasks that I presented are finalized to understand the mechanism of resistance. Then the project has also the objective to produce a new resistant germoplasm. At the moment can be done through conventional breeding. As you can see here, several crosses have been done during the past years, including at least one potential parental with resistant characters, so that we can hope that in the progenies we have new plants with these which harbor the resistance as the parental. The bottleneck is that we need to carry out this large breeding program. We need field plots, and we have to thank the Onlus Save the Olives, because they are collecting funds to and make available plots for the research. And these are also some of the plots that have been already realized to start to test this new germoplasm. This has been done in collaboration with a regional agency. And just in few words, this slide to summarize that this is a process to produce and to generate a new cultivar for olive. We need at least 12, 15 years. So there is a need for long-term research. This is the example in olive to register a new cultivar resistant to verticillium. In the case of Xylella is even more complicated, because to infect and to assess if a plant is susceptible or resistant to Xylella, it takes several years compared to what is done with verticillium. And so with this project that lasts only 3 and 4 years, we could not make all the program, but we are capitalizing all the previous investment in this direction on the resistant studies, so that we can advance the activity. We know that we will not reach at the end a new variety, the activity will be in progress, but and we have to thank for this program all the collaboration between private association and the research institute, because without having a large facility and plots available, it's quite difficult to carry out this activity, as well as several organizations in the Mediterranean countries which helped to collect the material. And I conclude with the information of the dissemination activity and the communication that you can find on our website. Thank you.
Thank you so much, Maria. Please, you can take a seat. And now we'll be hearing another project, Integrolive, with Professor.
Here. Good afternoon. Integrolive is a national project supported by the Italian Ministry of Agriculture. Is an interdisciplinary and multifaceted project that aim to develop an integrated approach to control as a main goal to control Xylella fastidiosa on olive plants, but also to act in the olive agro ecosystem to control also other olive pathogens. Obviously for an also economical point of view to reduce costs of plant disease control in the agro ecosystems. In the projects there are 4 research units. The university plant pathology group of my university, University of Molise, 3 National Council Research Institute, and the University of Bari, and also University of Naples. We have 7 work packages, and 4 of these from work package 2 to 6 are research work packages. And the first is the management of the project, and the WP7 is the activity that now we are made the communication and dissemination of results. The work package 2 aims to evaluate, to screen a large numbers of products, in general sustainable products, biological control agents, for example, natural compounds, commercial formulates of these products, but also experimental products. This activity, now we are at the 2nd year about of activity, and then this, the activity of this work package was concluded, and we have selected several products of various types, and these products are now evaluated in screen house, under quarantine screen house conditions, in the field, on different varieties of olives susceptible to Xylella fastidiosa, such as Oyarola and others, both on young plants, but also on old olive plants. In particular, in the infected area, we are conducting 3 experimental fields, 1 with young plants and 2 in different locations, and 2 on old olive plants. In the work package 3, the work package 3 regards the evaluation in planta on potted olive plants of products that we selected in the work package 2 in vitro and laboratory screening. And this activity is still in progress under greenhouse conditions. Contemporary in the VP4, we are studying the mechanism of action of the most promising products in regards to the activity on the pathogen, but also in the interaction with plants and in the multitrophic interaction plants, pathogen, and products. And also this activity is in progress, as well as the field trials that are conducted from 1 year in the field. As you know, the cycle, as also Maria said, of Xylella fastidiosa disease in plant is very long. And then we, in the first year, have applied several treatments, about 6-7 treatments of single products or also combined products in protocols, specific protocols. And now we are evaluating evaluating symptoms, but also the colonization of the bacteria inside the plants. And this activity is carried out on younger plants in a field where we are evaluating experimental products, but on all the mature plants we are evaluating commercial formulates of different products that we selected in the previous work packages. These activities are developing in cooperation with two assaying center, the assaying center of Basile Caravia, of this research center, and also the Biofarm of Ercolano, Napoli. Results, the experiment are in progress. We hope next year to acquire good results. The work package 6 involve a committee of experts of our team to set up, to choose the better protocols that we set up in the project in order to propose these protocols for practical application in the field, we hope in the close future. The work package 7 regards the dissemination and communication of results. For further information, we also have a website that you can visit to acquire more information about. An important aspect is that our protocols can be also very useful not only for susceptible varieties of olive plants, but also for tolerance, because with the new system of growing with plants more close in the field each other, we expect to have more impact of other diseases, fungal disease, but also other bacterial disease. And moreover, these new varieties, as the Lecchino, for example, and others, are tolerant but not immune to the Xylella. As you can see also here, some symptoms of recent symptoms on Lecchino that we are observing in the field. And we have to don't forget that olive tree is not for olive and oil production, but is also connected to history, culture, tradition, landscape of a territory as the Apulia region. We have a century and also plants that have thousands of years. The effective control of Xylella on olive plants is very, we are aware that is very, very difficult and an ambitious challenge. However, as Albert Einstein said, it is in the crisis that the best of each person, in our case researcher, emerges, because without the crisis, all wines are just gentle breezers. The Integral Olive project will most likely not solve the problem Xylella, but it will help to establish a sustainable coexistence in the infected area. Our project is addressed particular to infected area, between the pathogen and the olive trees. These are all the researcher colleagues that are participating on the project team, and also we give many, many thanks to our younger staff, our younger co-workers that are cooperating on all activity with very, very high enthusiasm. Thank you for your attention.
Thank you so much. Please take a seat. So you've now heard from effectively 4 projects, BIOVEXO, Omi Breed, Bexil, and Integrolive. So different projects, different approaches as well on how to tackle Xylella. And I would like then now to get a bit of feedback from you. If we can just show the audience how to connect. So what I want you to do is whip out your phones, get your phones out, and you can connect here to very quickly, or very soon. There will be a QR code, or you can go to a website called menti.com. Can we put it up? You have it? Yeah, okay. It'll be coming in a second. Here we are. Okay, so you have a QR code here, or if you're not a big fan of QR codes that then kind of track you to the bone, you know, they will track you wherever you go from hereafter, a little bit like Xylella in a way, you know, tracking you down. You can also go to menti.com and use the code 4800-1246. Is everyone good? I mean, you may not all be connected yet, but you're almost there. No problems? You're all in? Ah, I'm in the way. Yeah, look at that. Plenty of feedback. Great stuff. Okay, so as you connect, I want to invite you to a first reflection, right? I want you to imagine that you are, let's say, the European Commission, or at least an organization with a lot of money. Okay, are we ready? Can I get a yay if we're ready? Okay, is anyone not ready with a nay? No, okay, we're good. All right, let's do it. So, here we are. Next slide. No, it's not, you don't have it here? This should work for me. No? No? It doesn't show? Okay, yeah, go for it. You do it. Yeah, I disconnected from the internet, that's why. Okay, this is completely pointless. Okay, so I want you to imagine you're the European Commission and you have 100 million euros in your pocket, okay? And you have to decide where you want to put your research budget. Combat Xylella. Now, usually when I would ask a question like this, people say, oh, we need a bit of everything, you know? That's why I'm asking the question in this sense. Here, you can actually fund everything if you want to, but you only have 100 million euros. So what you need to do is to split your 100 into these 4 different options. You have genetic resistance and harnessing plant microbiome, you have integrated pest management, advanced detection methods, or biological controls. Now, don't think about it as how much will it cost. Think about it more in terms of what is a priority, okay? Because maybe you wouldn't put the same amount of money to different things because they just cost more or whatever, or the research is more complicated. Forget about that. Forget about the money in that sense. Take it as a prioritization exercise, okay? And I would like people to vote, like maybe go up to around 30 odd people. That would be good. We'd be around half, just to get a sense of where the audience sees the importance of these different aspects, because I think you've all touched upon some of them in your various projects. You've taken a direction, let's say, and I think it will be interesting to hear also from the audience on this. It looks like, for the moment, genetic resistance, integrated pest management are the top players here, and biological control and advanced detection methods a little bit less. Maybe a first reaction from Giuseppe, if you can. I think you took an approach that's more on integrated pest management, a little bit more holistic. You then have to sort of agree with coexistence. Could you maybe just comment then on this, like how much do you think integrated pest management is where you would also put your money, and how much do you think other aspects should be considered as well? And please click on the button. It's okay.
In my opinion, all strategies should be combined in an optimal way, because we, against a so serious problem, we have to use synergies of these means and strategies. For example, genetics, the genetic improvement is important, but also is important to adopt all practices to keep plants in a good condition of resistance, environmental resistance, other than gene resistance. The genetic improvements, we have, don't forget that plants is made by genes, but also the pathogen have genes. And then in the future, perhaps the situation will evolve. Mm-hmm. And then we can have biotypes of the pathogen that overcome this resistance, mm-hmm, that the genetics will develop. And then we have really other strategies to reduce risks that the plants can be again susceptible to the pathogen and attached seriously as for autochthonous varieties actually.
Yeah. And well, we can't see it now, but basically genetic resistance and integrated pest management are clearly the top two. Stefan, I mean, obviously you're working more on the biologicals aspect here, but you need to somehow connect to the genetic resistance as well, right? I mean, varieties will develop. How much do you think these two issues can combine?
We need to work on all these aspects. We cannot work individually only on one. All these research topics are necessary to develop to fight against Xylella. There was some talk saying not only one solution can reduce the disease, and we need several approaches. We need diagnostic tools to first detect the Xylella. We need some biocontrol agent, not only one, several one. We want to have more tolerant variety, but also to study the effect of our biocontrol agent on to have more tolerant of the tolerant variety. Also the plant microbiome, I'm working on that. And of course, IPM strategy, because you have seen also in BIOVEXO with curative approach and IPM strategy, we have different results. on the Xylella population. In the case of curative, there is no effect on the Xylella population, and on IPM, we have a reduction. So all these aspects are necessary to study, and honestly, it will be better to have 25, 25, 25, and 25% of each, because we are not expert on all these kind of things, but all together we can maybe change the situation in the future.
Yeah, and I mean, actually, it's not even quite a percentage. I mean, everyone's sort of placing it. It's a very tricky one, but I think it just gives a picture of the breadth. And if we get, let's say, most people thinking the priority is, let's say, genetic resistance. So to come back to Maria's projects as well, it's also probably the one that will take the most time. And that in that time, many other things can happen.
Yeah.
So how do you manage that in terms of the research to project yourself in what will only come in minimum, let's say 15 years, but by then maybe a lot of these other tools have developed and it's another kind of genetics that you need again?
One aspect I want to remark before answering your question is that, as I showed, the resistant plants are not plants immune to the infection, so they harbor the bacterium, and this means that if these plants are under other stresses, not well managed, or as Professor Lima was mentioning, being under multiple stresses, they can also express more severe symptoms than usual they do. So it is important, the IPM concept is also in this direction, that we need a resistant plant, but being a specific type of resistance, which is not total resistance against the bacterium, we need to combine with other strategies to maintain the plants in the best condition so that we have a resilient plant. It's not enough to have a resistant plant, but we have to grow the plants under the good condition. If the bacterium, if we are in an epidemic area where the bacterium continue to circulate and we have a pressure of inoculum. Regarding your question, it's important to say that maybe even at EU level, the concept on the genetic biotechnology and genetic transformation is a concept that start to be more accepted, and so we have some project which started to explore also in olive the new technologies for genetic improvement, which we, at the moment, we cannot forecast to develop new plants in short time, but we believe that once the technology are optimized in olive, we can shorten that period that for conventional breeding now is more than a decade.
And maybe, I mean, we touched upon this towards the end of the previous session on the interaction, right, between research and business. You want to add something? Please, quickly.
If I can add, obviously the biological control, in my opinion, is included in the integrated pest management. And for the last point, advanced detection methods obviously are very important, but we have to distinguish infected area from non-infected area. For non-infected area, advanced detection methods are very, very important in the view that preventive methods are better than curative methods when possible.
Yep. Okay. And thanks to everyone who participated. We will come back to these things, so don't worry. I know it's fun. But one thing I wanted to touch upon, because at the end of the previous session there was a bit of conversation around the interaction between research, business, uptake of research results, and how much research results can be shared. I would also like maybe a quick reflection on how the different projects have managed that interaction or are managing that interaction. How much are you interested in actually getting products in the market in the end, and how you think you can achieve that? I mean, Stefan, obviously that's a core part of the project because it's a kind of requirement in a way, or ideally. So I think, can you just say a word maybe about how you've managed that or how you intend to manage that?
Yeah, at the beginning, our goal was to help the farmer and to reduce the Xylella symptoms. So, as a scientist, in my opinion, we give a service for the society. And of course, we made a lot of publications, but not only publication, we need to have hand product to help the farmer with the situation. So it's both science and go to the final product from the basic knowledge to the end and to be on the market, in my opinion. But I'm working in an RTO, so I do all these kind of things.
And Maria, in your case, can you maybe comment a bit on the interaction, if any, with the business side?
Yes, in our case it's not exactly in the business side, but as I mentioned, the public and private partnership for this long-term project is quite important, because developing new variety does not mean that we have only to take care of the resistant characters of these plants, but also the agronomic feature of these plants, if these are then accepted by the consumer, by the producer. So the interaction with the producer to do a large-scale validation, and so to use their farm to do this activity is critical, it is essential, and so also for this in this line there is there are agreements to develop together these long-term projects.
Okay, thank you. Giuseppe, in your case, is there any interaction?
As before I evidenced, an important aspect is the economic aspects. And then all of these topics are important, but we have to consider that the costs of the farms that will be increased to control this unexpected pathogen in this last year. And then all new measures that I suggest for practical application, we have to need to perform an economical evaluation. Then in our project is different from others, because as goals also all pathogens of olives, together with the Xylella fastidiosa. And then when our products were evaluated also for side effects, also with fungal and other bacterial pathogens. And then when farmers apply a treatment, this treatment is not only specific for Xylella, but also for other pathogens. And then cost of control decrease. The economical aspects is very important.
Okay, thank you. Any questions, comments? No? All clear? All good? All right, so in the sake of time, I would like to then thank the three of you for having shared your views. We can give them a warm hand. And we will then move to the next part of this session. We will now be hearing from Gianni Giglioli. He's a research scientist at the University of Brescia, and we'll be discussing innovative strategies for managing Xylella fastidiosa. And after Gianni's presentation, I'd like to bring Maria and Pasquale back up on stage. Okay, so please welcome Mr. Gianni Giglioli.
Thank you. Good afternoon. I try to manage the complexity here because I see that now it's so much easy. Okay, let me briefly introduce myself. I'm not an expert in the Xylella biology. I'm a modeler, so I'm more interested and skilled in terms of analyzing data and interpreting the system dynamics. Although I think that my role in developing an IPM scheme is important, and I try to show why. What is IPM and how to develop IPM? Basically, is a matter of considering three steps. First of all, to revise the option for management. Second, to put them together. And third, to test them. So I try to go through the 3 steps, apart from the last one, because this is not part of the BEXIL project. That is the project in which I work. Okay. So, it comes. Ah, okay. So I anticipated the complexity and now I got it. Yeah, so, yeah, it is. This is to change the slide. Okay. That was my mistake, I'm sorry. This is for the change. I pushed that. What is the black screen? In italiano. No, per cambiare? Ho usato quello. Ho sbagliato, ok. So, why we are now in a condition of talking about IPM? Because it's in 2013 that we discovered Xylella here in Europe. Since then, a lot of phytosanitary effort putting together responses has been considered by EU in terms of defining quarantine measure to prevent new introduction, in terms of surveillance, monitoring activities, defining demarcated areas, and try to eradicate or contain Xylella in the area where we discover outbreaks. So, even if Xylella is still expanding, even if we have seen so much complexity in terms of the strains and the expansion in the spatial dimension, the status of Xylella is changing. And this is a common pattern for all the invasive species. So basically, we are in a transition between what is a so-called a PRA, pest risk analysis or assessment, depends on if you want to consider the management or not, to another status in which the key actors are farmers. So it's the transition between PRA and IPM that is now occurring, and now is the time for putting more attention to the IPM. What does it mean? It means to look at all the options that we have, put them together, and see what are the best combinations that you want to understand and test and apply. So many EU projects have been founded, and we have accumulated a lot of knowledge in terms of the biology of the vector and the bacterium, their interaction, the epidemiological pattern, but also information. We have seen the project today on BIOVEXO and the result they obtained in terms of option for managing both the bacterium and the vector. BEXIL project is a new EU-funded project that has a specific aim of putting together a multidisciplinary approach to fill knowledge gap, assess socioeconomic and environmental impact, and develop practical solutions to manage Xylella fastidiosa outbreaks in the EU. I'm responsible for work package 7 within this project. It is aimed at let's say, integrated the knowledge from the project, but also the knowledge accumulated outside this project in other experience and projects in the EU, but not only, and try to synthesize them and create a knowledge structure that can support the design of IPM schemes. And this is particularly the aim of the task 7.5 in the work package number 7. So, what is the methodology that we are following in our project for developing IPM schemes? Basically, a few definitions that is important to help you in terms of the words that I'm using. We distinguish between an IPM option that is a strategic, tactical, operational intervention with the aim of limiting the population abundance of the vector, the epidemics and the impact of the Xylella bacterium, considering the 3 classical phases of the prevention, monitoring, and control. An IPM scheme is a combination, a coherent, hierarchical, organized set of IPM options. But what is important here is an IPM scheme is context dependent. So we cannot talk about a general IPM scheme, but we have to define it in terms of all the aspects considering the legislative constraints, of course the crop type, the local management practices, the actors involved, the epidemiological status of the disease locally, and also socioeconomic aspects. So, following this definition, we are trying to put together a scheme summarizing all the options that are now known and available and somehow considered that are relevant or promising for managing Xylella. They are divided in 3 phases: prevention, monitoring, intervention. They were grouped in categories, and there is a non-compressed, there are many others probably, but they are more promising and the one that they are considering in the BEXIL project. Okay, starting from that, we asked the experts in our project, in the first phase, but this is just the first phase, so a second phase of expert judgment elicitation is ongoing, to judge the options that are known and are available, considering 7 criteria. From the level of knowledge, how the option is fully known from the scientific point of view, how we can judge the effectiveness, and so on, up to the level of readiness. So if we are able already to apply it or not. And according to the clear definition, we try to evaluate in a scale of 5 levels, how we consider each of the plenty of options in terms of the 7 criteria. So this is a preliminary results from the first round. The criteria were different, but you see from the different colors that we have already accumulated knowledge for making some selection and guiding us in defining schemes, putting together the options. Because it's clear that the efficacy that we expect from putting together the option is to consider an optimal combination of prevention, monitoring, and intervention, but also to clearly fine-tune the proposal to the specific epidemiological, economic, and social context. Okay? No, sorry. What we expect from the second round is a more sophisticated analysis in which from each option we will have the 7 criteria that will be judged in terms of the median of the value from 1 to 5, but also try to capture the uncertainty. So how is the agreement among the experts? If they agree, so we have less uncertainty. If they don't agree, we have higher uncertainty, and this is something that could influence us in terms of putting together this kind of option with others. So agreement among the experts is very important for the selection of the options. And then we have the most important steps. So the idea is to define 3 levels of complexity, that means effort, basically, combining different options for each of the 3 crops that we are considering in our project, so almond, grapevine, and olive. We expect having more than 1 scheme for each combination of complexity and crop, and this is exactly what we are doing now in this month, before the end of the project. It's not easy, because it's a matter of combining also the different views of the experts, and in particular try to translate the expert knowledge in numbers in order to help the selection of the most promising options. This is just an example. So we have crop is olive. We are in an endemic zone, not an epidemic, endemic. And we see the 3 levels, and this is a combination given by an expert that will be combined with other experts, and we will see how we are able to converge in a limited set of schemes. You see there is a gradient in terms of complexity and cost of implementation, and we try to save the important criterion of minimizing the risk for each level of complexity. This is another important aspect to be considered. A lot of knowledge can support the design of a scheme. For example, you see in the monitoring, the most simple, no monitoring, or vector monitoring in the higher complexity scheme. But this does not mean that you cannot gain information from the surrounding and from experience. So the problem is how to convey this information into a tool supporting the implementation of the scheme. And this is exactly what we are doing the next step. So in the project, we are developing a decision support system. What is a decision support? Oh, sorry. A decision support system is a tool that can be used by web, in which we put together a series of information and a way to, let's say, support a decision by the user. First, we have to define the scenario. Where is the production unit? What are the context-specific information to be considered? What is the crop you are dealing with? Who is the user? A farmer, a nursery, or also a risk manager with a question mark? And what are the intended use of this scheme? Do you want to just manage your crop because you know there is Xylella fastidiosa, or you are in a completely different condition? So, based on the definition of all these aspects, we define a scheme. This is a logical scheme, of course, that to be implemented in a software, in which we have a user guidance, and the most important, the decision support per se, so the most important part of the scheme, that is supported by a decision tree that depends on the context. According to the context, you define your objectives, and you are able to review the schemes and the options associated to the schemes that are already evaluated by the experts. But of course, you are also allowed to put together your options and try to bring the evaluation in order to have a pre-evaluation of what is your decision, is it good or not, with respect to what the expert said. Okay, this is an example of the leaflets that we are developing, supporting each scheme. So there is the first level, the information recommended for the specific context, the list of options, and general guidelines for the implementation of those options. Then there is other leaflets specifying further the information that is content here. And for example, okay, the first level describes all the options that are grouped, the expert evaluation is reported, and there are also supporting information for better understanding the options. And this is the final one in which more information for the specific option is given, but also supported by validated bibliography and web references. Okay, conclusions. So in the BIOVEXO project, we moved from the idea is that a lot of experts have accumulated a lot of experience, and nothing better than what is going in their mind in terms of synthesizing the knowledge, and nothing better than putting together all the experts and try to synthesize a common knowledge, also with differences. We can capture the differences and consider them. But then we have to translate this knowledge into operational tools, and this is because we are developing this decision support system. And finally, this is not part of the BIOVEXO project, so it's a matter of the following phases of evolution of the IPM schemes. Try to understand if this suggestion is supported by quantitative tools, models, or any kind of quantitative analysis, in terms of trying to preliminarily validate if the goodness of this combination. But finally, the third phase is the testing phase in field, in which the most promising schemes will be put in practice in terms of consider their effectiveness in specific epidemiological and economic contexts.
Thank you very much, Gianni. No, I've actually changed my mind. So what we're going to do, are there any immediate questions for Gianni? Yeah, please, I'll jump.
Wow.
Wow, that's athletic.
Yeah. Impressed.
Thank you very much for the presentation, very interesting, like others today. I have a question. I saw that in your prevention part, the vector is not appearing at all, while it's key to prevent the spread of the disease. Could you explain why?
Okay, on the same side, you can see I repeat the same measure in different columns. So in my view, it's better that we concentrate in terms of controlling as an intervention part for the vector. I don't have an idea how do you define a prevention for the vector. So vector is there, it's part of the biological community.
If the vector would be absent or at very low populations, then the likelihood of getting an infection is also very much reduced. So you could take preventive measures targeting the vector.
Maybe it's a matter of definition. I don't think that you can reduce the population without an intervention. Neither I think we can eliminate the vector within, from a natural community. And this is because I put in the intervention part.
Yes.
It depends on the perspective,
well,
probably.
Maybe we should have a bilateral chat.
Yeah, sure. No, but I agree.
I was just wondering what's your rationale behind.
No rationale. Unfortunately, for IPM, as you probably know, it is also a matter of definition. You can put the different options in different phases, as I have done in some part. For example, you will see repetition for the plants, for example. But in my view, controlling the vector as one of the components of the epidemics is part of the intervention. From the disease perspective, of course, it's a matter of prevention. But it's a nominal part, not so substantial part. We all agreed, I think, that the vector has a key role in promoting and sustaining the epidemiological dynamics.
Okay, do we have one more question for Gianni? We're good? Okay, so I'll invite you to come back here to the audience.
Don't jump,
No, you can go the other way. Yeah, we won't all start jumping here. And what we'll do, and also for Maria and Pasquale, we'll take the next part here. I'll actually just ask you to get back onto our little tool here. I just have a couple of questions for you, and I will ask then a few speakers to comment on that, and also to get your views. Okay? We'll do it from here. It'll be quicker and more efficient. So can we put the tool back on? And so we've heard a presentation that, okay, you're already going for it. You need the QR code? You need the QR code again? It's the same one. I've put the QR code up again if you need it. So we've heard of all the different intervention points that are possible. And again, I know that usually people say, yeah, we need all of it, and I know, but I always have to force an answer, okay? So here the idea is to ask you what is, according to you, the most critical intervention point for managing Xylella? Quali sono gli interventi più critici nella gestione della Xylella, okay? Thanks, Pasquale, for the translation on that one. So you have vectors control, quarantine measures and prevention, control of host plants, resistant varieties, or interactions with the endophytome. Now this is a ranking, okay? It's a ranking exercise. So again, it's about where you see the most critical intervention point from 1 to 5, okay? And you've heard already many, so this is a reflection moment. And I want you to take it, okay? Because I've been pushing you all day to listen and be attentive, and things are going fast. Now you have to think for a second. With some obvious interruptions on the thinking, but it's okay. All right, and I will now gradually go to some of my speakers to get a quick reaction. So I would actually like you, Maria, to give us your ranking and your comment on what you see.
I have some difficulties because they are the application of this measure have a different level of application. So vector controls is at farm levels, whereas the quarantine measure is something that affect the phytosanitary authority, the legislators, and so on. So to put on the same level for me is a little bit difficult. So it depends on the situation, but for sure, based on the experience we had here, the application of the quarantine measures have been quite well debated and had different view because of course quarantine for plant means the removal of the plants, and in case these are valuable plants like olive trees, has been something really critical to put in place and to accept and to implement.
From the experience of BIOVEXO vector control. So vector control is a critical point, I think. The other, as Maria said, apply in different contexts. So prevention is important in diagnosis or are important for the buffer zone to avoid that the bacteria, to avoid the spread. While resistant varieties are important to replace the people, to restore the cultivation of olives in the endemic area, as they say. So vector control, however, to me is the critical intervention point.
Thank you. Maybe also a reaction from Gianni on the results that you see here, and what would be your comment?
So the first thing is that I see that quarantine measure and prevention probably also consider part of the others. No? I also think that the vector control is a key factor. I would put the quarantine measure at that stage probably in the third place. So because also resistant varieties, as in the previous pool that we have done, was regarded as one of the most promising activity. So I see we are combining different views, and probably I'm still in favor of considering vector control one of the most important ones.
Thank you. Would anyone else like to react to what you see, or disagree or agree with the outcome here, or would like to share a view on what you see here? Anyone want to share a comment? No? Does everyone agree with this ranking? By and large? Yeah? Okay. Is anything missing? No? All right. Okay. All good. So what we'll do now is have one more, which will be of interest, I think, to Karen, potentially. Let's see. The next question is Europe in a more difficult situation with managing Xylella than other regions in the world due to its more restrictive approach to insecticides? Because obviously there are also other parts of the world where Xylella is present, not just in Europe. So let's get your views on this one. And finally, you have a definite answer. You know, you can go for one or the other. You don't need to rank. You don't need to think this through too much. You just pick one. In fact, you can only pick one. So I'm going to love to get a little reaction from Karen on this one. No, but still, you're with the European Commission, so you may have a view on this. All right, so clearly there seems to be a very definite yes on this question coming through. So may I get a first reaction from Karen on this one?
I will pass the information to my colleagues working on pesticides.
Okay, no, but that's an impressive, definite yes. Anyone would like to comment on that? Anyone share a reaction, a comment? That's a definite yes. I'd like to hear why. No? Anyone? No? No, no, no? No comment? It's a clear answer. Okay. Well, tell us why.
I voted partly yes, because otherwise BIOVEXO does not make sense. So it's normal. So if we rely on only on insecticide as a miraculous problem solving, so we have no other.
I have a question to everybody who answered yes. Because Xylella is a bacterial disease, but you say it's about insecticides. So shall I understand then the story is about a vector control? I mean, could someone maybe share the thoughts about that?
If there is no vector, there is no Xylella transmitted. That's all. Sorry.
Come on. We're getting into the core here. We're getting into the core. Anyone else want to react to this? Come on. I know I can get one more reaction from you. Come on. Is this what we're talking about? Vector control? Because the comment from Pasquale is a good one, right? It's not all about insecticides either.
So comments, if there is no restriction to some common insecticide we have used by the past, well, nefast for the environment, we don't solve the problem. There is many problems in that. And especially in Europe, some countries, they say, okay, we will stop this insecticide in 2030, and finally, it's like, okay, we still authorize a bit because we don't have other solution.
We're just displacing a problem to something else. No? No other thoughts, comments on this one? Okay, so it's a little bit more nuanced than what would first appear here. You know, has anyone felt maybe I shouldn't have voted definitely yes following this brief interaction? Would anyone change their mind? Come on, I know there's someone who would have maybe changed their mind, even if it's just going from definite to partly. No? Okay, I will let you free. You have now earned the right to have a short break, coffee, okay? And then we'll be back at 4 p.m. and we'll be discussing the business side of the project, okay? So see you back at 4 p.m.
Thank you.